In the gentle stillness of night, when the world has softened into quiet edges and slow breaths, it can be comforting to remember that the story of the human brain is far older than any moment we experience within it. The mind that listens now, settling into a comfortable rhythm, is the product of an ancient journey, a journey that spans oceans of time, long before memories, long before language, long before even the simplest creatures found their way through the world. To begin this documentary, it helps to imagine the Earth not as it is today, but as it once was, a young, restless sphere wrapped in clouds of steam and warmth, its surface shifting with volcanic light, and its oceans just beginning to gather in the hollows of the cooling crust. This world was both familiar and alien, holding the early conditions that would one day allow minds to grow, sense, feel, and reflect. As these early oceans settled into a quieter rhythm, something subtle began to occur within them. Simple molecules, carbon, hydrogen, oxygen, nitrogen, drifted in the warm waters, pulled into new shapes by gentle currents and the faint energy of sunlight. These molecules were small and unassuming, yet they would eventually become the foundation for living cells. There was no hurry in this process. Nature worked slowly, letting time do the shaping. Over millions of years, these molecules began to form more complex arrangements, folding and linking into patterns capable of storing energy, breaking down nutrients, and forming boundaries. From this long period of molecular wandering came the first cells, small pockets enclosed by delicate membranes. Inside these first cells, information moved in simple patterns. Chemical signals drifted through the interior like tiny messengers, activating reactions or quieting them, helping the cell decide how to respond to the world around it. These early forms of communication were so basic that they hardly resemble what we now call signaling, yet they contained the essence of what would eventually become the intricate electrical impulses of the nervous system. Even here, in these calm early moments of life's story, the first faint traces of what we might one day call a mind were beginning to take shape, not as thoughts or feelings, but as the simple ability to sense and respond. As time continued its steady work, these first living cells began to join into larger communities. Sometimes they remained individual, drifting alone in the water, but in other moments they came together, forming filaments, clusters, and early colonies. These arrangements allowed them to share tasks, making life a little easier. Some cells specialized in movement, gently pushing the colony forward. Others took in nutrients. Some focused on responding to the outside environment. This quiet cooperation marked a new stage in life's unfolding, the rise of multicellular organisms. These early organisms had no brain, no central processor coordinating their activity. And yet, within their shared body plans lay the seeds of something more sophisticated. Coordination created new demands. To act as a whole, these organisms needed better ways for cells to communicate, sending signals not just from one side of the cell to the other, but across an entire body. In these ancient seas, something new appeared: cells that were especially good at carrying messages. These were the earliest precursors of neurons, long before the word neuron existed. Over time, these cells developed thin projections that could reach farther, connecting one group of cells to another, allowing signals to travel with greater speed. At first, these projections may have been simple, carrying only the faintest chemical clues from one area to another, but gradually, driven by slow evolutionary pressures, these messenger cells grew more refined. They became capable of transmitting impulses and responding to them, creating an internal network of communication that was more organized, more precise, and more adaptable than anything that had existed before. The development of these early signaling cells was a quiet revolution. With the ability to transmit messages more quickly, early organisms could coordinate their movements with greater efficiency. They could adjust their shape, their feeding patterns, or their responses to danger. Over time, these connected networks formed what we might call nerve nets, simple webs of communicating cells that covered the organism's body. These nets allowed early animals to respond more fluidly to the world around them, curling or expanding when touched, drifting more deliberately through the water, or capturing food with graceful, coordinated motions. There was still no brain at this stage, but the groundwork had been laid. As we begin our journey into the evolution of the human brain, it is important to hold on to this sense of slowness and accumulation. It emerged gradually, shaped by the needs of living things over immense stretches of time. Even the most complex human behaviors, memory, imagination, empathy, are built upon layers of simpler structures that trace back to these earliest creatures. In the stillness of this opening part, imagine drifting through those warm, ancient oceans, witnessing the birth of biological communication and understanding how something as delicate as a thought is tied to these early chemical whispers. The same processes that once allowed a primitive organism to sense a shift in water pressure or the presence of light now support the intricacies of modern human experience. They allow neurons in the visual cortex to interpret color, neurons in the hippocampus to store memories, and neurons in the prefrontal cortex to guide decisions. Life, the uploaded files remind us, is built from layers, from atoms to molecules, from cells to tissues, from tissues to organs, and from organs into complex systems that coordinate the entire body. Among these systems, the nervous system stands out for its elegance and its ability to unify action, sensation, and thought. It gathers input from the world, integrates that information, and coordinates the body's response in a seamless flow. This hierarchy, which defines much of biology, is reflected in the structure of the brain itself, small circuits forming larger networks, which in turn create broader regions responsible for intricate functions. As we move deeper into this documentary, we will explore how these early nerve nets gradually became centralized, eventually forming the first brain-like clusters. We will see how sensory organs encouraged expansion, how movement required coordination, and how the slow unfolding of evolution sculpted the remarkable organ that resides within the human skull. But for now, in this opening moment, allow yourself to rest in the awareness that the story of the brain is not a sudden event, but a long, unhurried river of time and transformation. This journey began with warmth, water, and the simplest forms of communication, and it continues now, in the quiet rhythm of your breathing, in the gentle settling of your thoughts, and in the ancient biological echoes within every cell of your mind, to Earth in its earliest moments. In the soft imagination of deep time, long before the world took on the familiar contours of continents and coastlines, Earth existed as a young and restless planet, wrapped in warmth, vapor, and constant change. To picture these early moments is to enter a landscape without quiet, a world shaped by slow cooling and sudden eruptions, where the air shimmered with volcanic gases and the surface glowed with the leftover heat of planetary birth. Yet even in this wild and shifting place, conditions were beginning to settle into the delicate balance that would eventually make life possible. The Earth of billions of years ago was not peaceful in a conventional sense, but in the gentle storytelling of sleep, we can imagine this ancient world breathing in long, steady pulses, preparing itself for the emergence of something remarkable. As the Earth cooled across tens of millions of years, the first oceans formed in broad, steaming basins. Rain fell for ages, centuries upon centuries of warm storms, until enough water gathered to smooth the planet's once jagged crust. These early oceans were not the clear blue expanses we know today. They were thick with dissolved minerals, warmed by geothermal heat, and stirred constantly by tides far stronger than modern ones. Moon and Earth were closer then, and their gravitational dance pulled the waters with a force that created tides as powerful as entire storms. Through this churning, the early seas became a giant mixing bowl, blending chemicals into new arrangements, supporting reactions that had never occurred before on the surface of any world. In these vast and shifting waters, the building blocks of life began to collect. Carbon, hydrogen, nitrogen, and oxygen elements, forged in the hearts of ancient stars, combined into simple molecules. These molecules did not appear suddenly or dramatically. Instead, they drifted, joined, separated, and reformed in countless patterns. Lightning flashed through the thick atmosphere. Heat from volcanic vents rose through the ocean floor. Sunlight filtered gently through the water, offering energy in soft, broken rays. All of these forces contributed to an unseen chemistry that had no specific direction, no intention, and no final goal. Yet slowly, across generations of reactions, molecules grew more complex, becoming chains and rings and spirals that could store energy, fold into shapes, and interact with one another in new ways. There is a quiet beauty in thinking about these early steps, because they remind us that life's beginnings were neither chaotic nor orderly, but something in between. The world was dynamic, yet patient. Conditions were unpredictable, yet favorable in just the right ways. The atmosphere lacked oxygen, so the earliest molecules existed in gentle balance, never burning away, never breaking apart too quickly. On the ocean floor, hydrothermal vents released warm minerals and chemical gradients into the surrounding water. These gradients acted like invisible pathways for energy, allowing certain reactions to happen more easily. Some scientists believe that life began here, in these sheltered pockets of warmth where chemistry flowed steadily. Elsewhere in the ocean, sunlight provided another source of energy, fueling reactions in shallower waters. The early Earth was rich with opportunity, and in this slow, simmering environment, the first primitive membranes began to form. These membranes were simple boundaries, thin layers that separated one set of chemicals from another. At first, they may have been fragile, breaking and reforming constantly, but over time, they trapped certain molecules inside, creating tiny, enclosed environments, protocells, where reactions could occur in a more focused way. These protocells were not alive, at least not in the way we think of life today, but they represented a transition, the moment when chemistry began to behave like biology, forming units capable of maintaining their structure and subtly changing over time. As the oceans continued to calm and swirl in great slow cycles, these protocells grew more complex. Others found ways to copy certain molecules, creating crude patterns of inheritance. Over millions of years, these early structures became the first true cells, tiny pockets, capable not only of surviving but also of responding to their surroundings. They could take in nutrients, expel waste, and maintain a delicate internal stability, a state known, even today, as homeostasis. This stability, as described in your uploaded materials, is one of the fundamental features of life. The ability to maintain balance, even when the environment changes. To imagine the beginning of life is to imagine the beginning of awareness, though not awareness in any conscious sense. These first cells had no nervous system, no senses as we understand them, and no ability to think. And yet, they reacted to light, to temperature, to chemicals in the water. They moved slightly when conditions shifted. They reorganized themselves in response to energy. These reactions were mechanical, but they were also the earliest hints of sensation, the smallest possible precursors to what would, billions of years later, become the sensory pathways of the human brain. For a long time, these first cells existed alone, drifting through the oceans as singular entities. But nature tends toward cooperation as often as it tends toward competition, and eventually, some cells began to cluster together. Perhaps certain environments favored clumps that could withstand currents better. Perhaps molecules on their surfaces made them sticky, encouraging them to remain close. Whatever the reason, though simple and small, they represented a profound shift. Now, life could become larger, more structured, and more adaptable. Within these early bodies, cells began to specialize. Some focused on absorbing nutrients, Others concentrated on movement. Still others became sensitive to environmental changes. This specialization created a new challenge: communication. To coordinate their actions, these early clusters needed a way to transfer information more efficiently than simple diffusion. Over time, certain cells developed the ability to transmit signals more directly. These signaling cells would eventually become the ancestors of neurons, though in these early organisms, their function was modest. They helped guide movement, regulate simple behaviors. Even in this primitive form, they represented the first glimmers of a system that could one day gather information, interpret it, and respond with precision. The Earth itself continued to shape life during this time. Its rotation slowed, its surface cooled, its continents began, faintly, to appear as landmasses. Weather patterns softened, the atmosphere slowly transformed, influenced by the earliest photosynthetic organisms, which released small amounts of oxygen into the air. This oxygen did not accumulate quickly. Instead, it settled into rocks and oceans, binding with minerals and slowly altering the chemistry of the world. Only after vast lengths of time did it reach levels that supported more energetic forms of life. Yet even in its earliest presence, oxygen allowed cells to develop new metabolic pathways, making it possible for them to grow more complex and powerful. In the stillness of this part of our journey, sudden dramatic events, but with the slow patience of planetary time, the Earth that would one day support dense forests, intricate ecosystems, and conscious minds began as a world of fire and steam, gradually cooling into a cradle for possibility. And within this cradle, the first cells formed the foundation for every living thing that would follow. Their cautious movements, their simple reactions, and their growing complexity set the stage for the long evolution of the nervous system. As we move forward into the next part of our journey, we will explore how these early cells began to cooperate more deeply, creating structures and communication systems that pulled life toward greater coordination. From these quiet beginnings, the first hints of a true nervous system began to appear, leading slowly and gently toward the emergence of the earliest minds. 3. The first cells and the birth of internal signaling. In the quiet unfolding of Earth's early history, once the first true cells had gained the ability to persist in their delicate membranes and maintain their inner balance, a new chapter in the story of life began. These cells, adrift in the warm ancient seas, were far from simple, despite their microscopic size. Within them, chemical reactions formed intricate patterns, each responding to the slightest changes in temperature, light, and nutrients. Though they had no nervous systems and no specialized structures, they held within their tiny boundaries the earliest whispers of what would one day become the foundation of sensation, communication, and thought. To rest gently with this idea is to remember that even the most sophisticated human experiences have their roots in these small and ancient worlds, where life existed without noise, without hurry, and without form beyond a thin membrane separating inner and outer. The earliest cells lived solitary lives at first, floating freely, taking in molecules that drifted by, and releasing others in return. They relied on diffusion, the slow movement of particles from one place to another, to communicate internally, and when necessary, externally. But this slow form of movement, while sufficient for simple life, carried limitations. As cells encountered more complex environments, they needed ways to adjust their behavior more intentionally. A cell's surroundings could change at any moment. The temperature might rise, the water could become more acidic, a nutrient might appear or disappear. To survive, a cell needed to respond not just passively, but actively. It needed to sense, interpret, and act. Inside these early cells, the first internal signaling systems began to appear. These systems were small networks of molecules that interacted with one another like tiny messengers. When something in the environment shifted, Molecules on the cell's surface detected the change and passed the message inward. Once inside, other molecules responded, triggering a cascade of reactions that allowed the cell to reorganize itself. These signals might cause the cell to move away from harm, shift its metabolism to use different nutrients, or prepare for division. These responses were slow and limited, but they represented an evolutionary turning point. Life was beginning to communicate with itself. This internal communication became more sophisticated as cells evolved. Proteins, the versatile molecules that carry out an astonishing variety of functions, began to take on specific roles in transmitting signals. Some proteins sat embedded in the cell membrane, acting like gatekeepers. When they detected a molecule outside the cell, a nutrient, a toxin, a shift in pH, they altered their shape. This change in shape acted like a signal, telling other proteins inside the cell that something was happening. The information then spread through the cell in gentle molecular waves, each reaction nudging the next into motion. This was the earliest form of a signaling pathway, and though far simpler than the electrical impulses that neurons would one day carry, it served as the essential foundation for all later forms of communication. Over generations, some cells developed more elaborate systems. They began to use ions, tiny charged particles, to create gradients across their membranes. A gradient is a difference between two sides, like a quiet tension. When the cell allowed ions to flow from one area to another, it generated small electrical shifts. These shifts were not yet action potentials, the rapid bursts of electricity that neurons would later produce, but they carried the same principle. Information encoded in movement, one side different from the other, flowing back toward equilibrium. Even here, in these ancient cells, the earliest faint vibrations of electricity existed, carrying the promise of more complex signaling systems to come. The emergence of these ion-based changes brought new advantages. Cells could respond more quickly and more precisely. Instead of relying solely on chemical messengers that traveled slowly through the cell's watery interior, they could alter their electrical balance in an instant, creating rapid internal communication. These small sparks of ionic movement were like the gentle beginnings of a language that life was learning to speak, a language based on differences, flows, and changes that could guide a cell's decisions. As life continued to evolve, individual cells discovered that cooperation could be more advantageous than independence. Multicellular organisms began forming, not because of conscious choice, but because of natural advantages. Clusters of cells survived better in certain environments, shared resources more effectively, and offered greater protection against harsh conditions. Once cells began to live together, they needed new ways to communicate across these small distances. Diffusion alone wasn't enough. Communication needed direction and purpose. Some cells became specialized for sending signals. These cells grew longer extensions that allowed them to reach other cells more directly. Though these early extensions were not yet true axons or dendrites, they represented an important step. They could pass information from one part of the tiny organism to another with less delay. These primitive signaling cells formed the earliest form of a neuron-like structure, a bridge between simple internal signaling and complex nervous systems. The inner workings of these early multicellular organisms began to resemble a quiet coordination. When one cell sensed a change, it could send signals to its neighbors, allowing the entire organism to react in unison. If a current shifted, or if light touched one side of the body, the cells could communicate this information across the cluster, adjusting movement or shape. In a sense, the organism was gaining the earliest form of a body-wide awareness, not awareness as we experience it, but a shared responsiveness, a unified reaction. This was the beginning of collective behavior, and it depended entirely on communication between cells. It is gentle and humbling to reflect on the fact that the nervous system, described in your uploaded material as the body's communication network, responsible for gathering input, integrating it, and coordinating responses, began in such tiny and fragile ways. The same principles that guide a modern neuron detecting changes, transmitting signals, coordinating action, originated in these early molecular interactions. What we now think of as complex processing began as simple cause and effect reactions, carried out by molecules passing information across microscopic spaces. The evolution of internal signaling also set the stage for another remarkable development: memory. In these early cells, memory was not stored as images, emotions, or impressions. Instead, it lived in the form of chemical changes. When a cell encountered a particular stimulus repeatedly, the proteins responsible for responding could become more or less sensitive. The cell, in a way, learned from experience, adjusting its internal pathways so that future responses would be quicker or more efficient. This rudimentary form of memory, chemical, quiet, and without consciousness, would one day allow complex organisms to store vast amounts of information, shaping behavior, guiding decisions, and supporting the intricate experiences of human life. As we rest in the slow rhythm of this evolutionary story, it becomes clear that the birth of internal signaling was not just a biological milestone, but the beginning of a long and delicate process that allowed life to sense, respond, and eventually understand. It all began with tiny shifts in chemistry, with the movement of ions across membranes, with the slow growth of molecules that could carry information. From these beginnings came neurons, nerve nets, spinal cords, and eventually brains capable of reflection, imagination, and self-awareness. In the quiet waters of early Earth, the first cells learned the ancient language of signaling, and from that language grew the entire symphony of sensation that defines the living world. With this gentle foundation in place, the next stages of evolution would shape these early communication pathways into organized systems, systems that would one day blossom into the first true nervous networks, guiding organisms through their environments with newfound coordination and purpose. 4. Multicellular life and the need for coordination. In the long, unhurried flow of Earth's early history, once the first cells had established stable ways of sensing and responding to their surroundings, evolution began to explore a new possibility: the joining of many cells into a single living body. This transition from solitary existence to communal life did not happen suddenly. It unfolded across millions of quiet years as conditions slowly shifted. Yet even with all this time, the emergence of multicellular life was an extraordinary change, one that required cells to learn how to live together, share responsibilities, and eventually coordinate their internal activities in ways that would set the stage for the nervous system and the earliest forms of organized behavior. When life existed only in the form of single cells, the challenges it faced were limited. A solitary cell needed to find nutrients, maintain its internal balance, and replicate. Its world was small and immediate. Whatever floated nearby could be absorbed. Whatever threatened it could be avoided or endured through simple movements or metabolic shifts. But as certain environments grew more complex, single-celled organisms discovered that living in groups offered unexpected benefits. Clusters of cells could create physical protection, shielding their inner members from harsh conditions. They could absorb more nutrients and share them. They could remain anchored to certain surfaces in turbulent waters. Most importantly, they could perform multiple tasks at the same time, something no single cell could accomplish efficiently. The earliest multicellular organisms were simple clusters, cells that remained attached to one another after dividing. These clusters might have begun as accidental groupings, mere strings or spheres of cells held together by weak bonds. But over time, natural selection favored those that could maintain these connections more reliably. Clusters that stayed together had an advantage in stability, allowing them to weather the shifting underwater currents. They also began to develop gentle internal divisions of labor. Some cells took on the work of capturing nutrients from the environment, while others focused on reproductive tasks. This division, though modest in its earliest stages, marked the beginning of cellular specialization, a major evolutionary innovation that would echo through all later forms of life. These clusters expanded and became more organized. The need for communication became increasingly important. In a single-celled organism, the response to the environment happened within one enclosed space, but in a multicellular form, information had to travel from cell to cell. If the outer layer sensed a change in temperature, nutrient levels, or chemical signals, this information needed to reach cells deeper inside the organism, especially those responsible for regulating metabolism or preparing for cell division. Without a way to coordinate their actions, the cells within the cluster would function independently, creating conflict, inefficiency, or instability. Evolution answered this challenge by expanding the internal signaling systems that were already present in single cells. Chemical messages became more specialized, traveling between cells in patterns that allowed distant parts of the organism to communicate. These messages moved slowly at first, diffusing from cell to cell, creating waves of information that guided shared responses. They developed longer, thinner extensions that reached out like delicate threads, allowing them to pass information directly rather than relying on passive diffusion. Though these threads were far simpler than axons or dendrites, they represented the earliest attempt at forming purposeful communication pathways, small bridges that allowed cells to coordinate with one another across the growing space of the organism. In these early multicellular bodies, coordination also required a greater sense of spatial awareness. A cluster of cells needed to know where its outer surface was, where nutrients were coming from, and where it needed to respond. This awareness was not conscious, but structural. Cells organized themselves into layers, each performing distinct functions. The outer layer, exposed to the environment, became more responsive to external cues. The inner layers specialized in protection, storage, or reproduction. This simple layering created a physical map, a basic geometry that shaped how the organism moved and behaved. Movement itself became a new challenge with the appearance of multicellular life. A single cell could swim or glide using its own surface structures, but a multicellular organism had to coordinate the movements of many cells at once. Some cells developed the ability to contract slightly, while others produced gentle currents that moved fluid around the colony. When these cells acted together, the entire organism shifted in a slow, unified motion. This was one of the earliest forms of coordinated movement, a precursor to the much more sophisticated motor systems seen later in evolution. Yet even at this primitive stage, coordination depended on communication. The contracting cells needed to know when to act, and the cells responsible for sensing changes needed a way to alert them. Over time, natural selection favored organisms that could respond more quickly and more effectively. Those that developed efficient signaling systems had better chances of survival. Some organisms created junctions between their cells that allowed ions and small molecules to move rapidly from one cell to the next. These junctions allowed electrical and chemical signals to pass in coordinated waves, giving the organism a shared sense of timing. When one side of the organism encountered a stimulus, a change in light, pressure, or nutrient availability, the signal could spread across the entire body, allowing a unified response. This was perhaps the most important step toward the eventual evolution of nerve cells and nerve nets. As multicellular organisms grew more complex, specialized cells for sensing the environment became increasingly advantageous. These sensory cells were early ancestors of what would later become sensory neurons. They were more sensitive to light, vibration, or chemical gradients, allowing the organism to detect subtle changes in its surroundings. Their signals had to reach other cells that controlled movement or internal processes. Slowly, a division emerged between cells that sensed and cells that acted. This division laid the foundation for the sensory and motor roles that characterize modern nervous systems, where some neurons gather information while others carry commands to muscles or glands. The uploaded materials describing systems of the human body emphasize that organs must coordinate with one another and that systems interact constantly to maintain stability and function. This principle applies even here, in these ancient organisms, where emerging cell types needed to work together as parts of an early biological system. No cell could act independently without affecting the whole. Coordination, cooperation, and communication thus became essential traits, just as they are in the organ systems of modern animals. Eventually, certain multicellular organisms evolved the first true nerve nets, simple networks of communicating cells that wove through their bodies like delicate threads. These nets allowed signals to travel with greater precision and speed, enabling the organism to contract uniformly or change direction more effectively. These early nerve nets were not brains, not yet, but they marked the first major step towards centralized control. They represented an early attempt by nature to gather information in one place, interpret it, and distribute instructions across the body. As we rest in the gentle understanding of these developments, it becomes clear that multicellular life represented not just a new chapter in evolution, but a profound shift in the nature of existence. Cells learned to live together, to share tasks, and to communicate in ways that made unified action possible. From these quiet beginnings arose the earliest forms of organized behavior, coordination, and environmental response. And from these same beginnings, the blueprint for the nervous system slowly began to emerge, setting the stage for nerve nets, centralized structures, and eventually, the first true brains that would shape life in remarkable and intricate ways. The story continues from here, as early nervous systems begin to develop more structured forms, allowing organisms to navigate, to sense, and to move through the world with growing complexity. 5. The evolution of the first neurons. In the soft continuity of life's unfolding story, after multicellular organisms had begun learning to live together and to share the responsibilities of survival, evolution moved gradually toward one of its most elegant and transformative inventions, the neuron. These early organisms, drifting quietly through the ancient seas, were becoming increasingly complex. They needed faster ways to respond to the world around them, more coordinated ways to move, and more precise ways to communicate across their growing bodies. Simple chemical signaling, which had been sufficient for single-celled life, no longer met the demands of these expanding forms. Diffusion was too slow. Localized reactions were too limited. Something new was needed, something that could send information swiftly and reliably from one part of the organism to another. Out of this gentle, persistent pressure, the first true neurons began to emerge. To imagine this moment is to picture simple organisms floating in warm, mineral-rich waters. They had no eyes, no ears, no centralized systems, yet their survival depended on sensing even the faintest changes in their environment. When light shifted, or when a chemical pattern drifted through the current, the organism needed to react. Some cells within these early bodies became more sensitive than others, specializing in detecting subtle environmental cues. These sensory specialized cells gradually gave rise to a new kind of cell, one that not only sensed, but also passed information onward. This passing of information was the beginning of neural communication. These early protoneurons developed elongated shapes, stretching their structures outward like thin filaments. These filaments allowed them to reach distant cells, not just neighboring ones. At first, these extensions were simple, offering slightly faster communication than passive diffusion, but over generations, the extensions became longer, slimmer, and better adapted for carrying signals efficiently. These early projections weren't yet true axons or dendrites, but they served the same quiet purpose: to connect one cell to another through direct pathways, reducing the delays that would otherwise hinder coordinated behavior. Inside these emerging neurons, important molecular changes were taking place. Life had already begun experimenting with ion gradients, differences in charge between the inside and outside of the cell. These gradients created small reservoirs of potential energy, and when specific molecular gates opened, ions could rush in or out, producing tiny electrical shifts. Over time, these shifts became more refined. Certain cells developed proteins that made the movement of ions more controlled and more predictable. This ability to manage ions, especially sodium and potassium, became one of the defining characteristics of neurons. As your uploaded materials describe, neurons eventually gain structures such as the axon hillock, where signals begin, and ion channels that allow impulses to fire rapidly. But at this early stage of evolution, these mechanisms were simple, only a faint prototype of what would come later. Yet even these faint sparks represented a profound leap forward. Electrical signals could travel much faster than chemical diffusion, allowing information to move across the organism's body with remarkable speed. For the first time in Earth's history, living beings could respond to changes in their environment almost instantly. With this new capability, early organisms gained a distinct advantage. If a current suddenly shifted, they could adjust their movement. If a predator approached, even in the simplest form, they could contract swiftly or alter their orientation. The world around them, always dynamic, always fluctuating, could now be met with coordinated responses that increased their chances of survival. As these neural cells spread through the organism's body, they formed interconnected networks. But as evolution continued its slow, patient work, the networks became denser and more intricate. These early neural connections began to resemble webs, giving rise to the first true nerve nets. These nets did not have central control. There was no brain yet, no central processing center. Instead, signals moved freely across the network, triggering local responses that rippled outward. In animals like ancient jellyfish relatives, this kind of diffuse network allowed them to contract rhythmically, propel themselves gently through the water, and respond to contact from any direction. The development of these nerve nets marked the moment when life first achieved coordinated movement at a scale beyond individual cells. Sensory information could travel across the organism, allowing it to act as a whole. If one part of the body encountered a stimulus, the entire organism could react in fluid unity. This coordination brought new survival advantages, and natural selection began to favor those organisms whose neural networks were more effective, more responsive, and more interconnected. Meanwhile, within individual neurons, the internal machinery for communication was becoming more sophisticated. Small vesicles formed, capable of storing chemical messengers known as neurotransmitters. When an electrical impulse reached the end of the cell, these vesicles released their contents, transferring information to the next neuron or to another cell type entirely. This chemical handoff added another layer of precision. Electrical signals could carry information quickly across the neuron, and chemical signals could pass that information to the next cell in a controlled, refined manner. This dual system, electrical within neurons, chemical between them, became the universal language of the nervous system. And even today, it remains the basis for all thought, movement, and sensation. Evolution continued to refine this communication. The extensions of neurons differentiated into two primary types. Some became dendrite-like structures, branching out to receive signals. Others became more like axons, long and straight, designed to send signals efficiently to distant parts of the organism. This division of labor allowed neurons to connect in more elaborate ways, forming circuits that could perform increasingly complex tasks. A signal could now travel from a sensory region through an internal pathway and arrive at a region responsible for movement or contraction. Step by step, the earliest neural circuits appeared. These circuits created a sense of flow within the organism, a gentle rhythm of incoming and outgoing signals that allowed for smooth, coordinated action. Movement became more graceful. Responses became more nuanced. Instead of reacting only to immediate contact, organisms could now orient toward favorable conditions, adjusting their behavior across a wider range of possibilities. The earliest forms of behavior emerged in these patterns, moving toward light, avoiding harmful chemicals, or drifting toward nutrient-rich regions of the sea. It is soothing to reflect on the idea that the human brain, with its billions of neurons forming intricate networks, grew from these ancient beginnings. The neurons described in your materials, cells with long axons, delicate dendrites, and synapses where neurotransmitters pass between them are the descendants of these early signaling cells. The basic structure, though now vastly more complex, retains the same elegant principles: electrical impulses, chemical transmission, and a constant interplay of excitation and inhibition guiding every response. As the ancient oceans continued to cradle life, the earliest neurons became the threads through which organisms touched the world. They formed the foundation for sensation, perception, and action. They allowed life to reach outward, not merely reacting mechanically, but responding with growing sophistication. They carried the first signals that would eventually lead to memory, thought, and consciousness. By the time nerve nets became established across multiple early species, the groundwork for more advanced nervous systems was already quietly forming. These primitive networks, simple as they were, demonstrated that communication could be organized, coordinated, and refined. They showed that life could connect itself internally in ways that opened new evolutionary pathways. In this gentle moment of reflection, we can appreciate how the evolution of the first neurons transformed the living world. What began as molecular whispers inside single cells grew into coordinated electrical messages traveling across complex bodies. These early neurons were the beginning of the long journey toward the human mind, carrying the first sparks of the complex inner life that now listens, breathes, and gently drifts toward rest as the story continues into deeper layers of time. 6 nerve nets, the earliest nervous systems. In the gentle hush of evolutionary time, after the first neurons had begun to stretch their delicate threads through early organisms, an entirely new way of living emerged in the ancient oceans. These oceans, warm and softly illuminated by filtered sunlight, cradled life forms that were slowly learning to coordinate not just individual cells, but their entire bodies. The appearance of neurons had granted them the ability to send signals rapidly, but the true transformation came when these neurons connected into networks simple, yet astonishing innovations known as nerve nets. These nets marked the first genuine nervous systems on Earth, and though they lacked the complexity of brains, they laid the essential groundwork for every nervous system that would follow. To imagine a nerve net is to picture a web of fine communication lines woven loosely throughout the body of an early creature. There was no central hub, no higher command center, no division between front and back. Because these animals had no distinct head or directional orientation, their bodies were often symmetrical in all directions, radiating outward like ripples on water. This structure suited their nerve nets perfectly. Signals could travel in any direction, reaching muscles, contractile cells, and sensory structures wherever they happened to be. This decentralized design allowed organisms to respond to contact or light from any angle, making their reactions fluid and immediate. The earliest nerve nets likely appeared in simple animals resembling the ancestors of modern jellyfish, sea anemones, and corals. These creatures drifted gently through their environments, or rested anchored to ocean surfaces. Their needs were basic: to capture food, to withdraw from danger, and to maintain their structure. The nerve net gave them a coordinated way to do all of these things. When a tentacle brushed against prey, the signal traveled through the net, triggering a wave of activity that helped move the tentacle, contract the body, or activate stinging cells. Everything happened almost simultaneously, not because of a complex brain, but because the net allowed signals to flow naturally across the body. This style of signaling created behaviors that were simple but graceful. Instead of moving with the focused intention of later animals, these early creatures moved in rhythmic patterns. A stimulus on one side could generate a full body contraction, pushing the animal slightly through the water. Repeated in rhythmic succession, these contractions became a kind of pulsation, slow, undulating motions that carried the creature forward. These gentle movements are still visible in modern jellyfish, whose pulsating drifts echo the ancient rhythms of the first nerve nets. The nerve nets did not operate with precise pathways like later nervous systems. Instead, they functioned as diffuse webs in which signals spread outward in circular patterns. This spreading resembled ripples flowing outward from a drop of water, and depending on the density of neurons and the sensitivity of surrounding cells, these ripples could guide behavior across the organism's entire body. Even without a controlling center, the net created order out of simple rules. A signal arrives, it spreads, and it triggers a coordinated response. One of the remarkable features of nerve nets is that they demonstrated, for the first time, the power of distributed processing. There was no single point of failure. If one part of the net was damaged, signals could still travel through other routes. This resilience was essential for early animals living in environments where damage was common. Small predators, shifting currents, and abrasive surfaces could harm parts of the organism. But the net allowed communication to continue. Even broken, these primitive systems could still orchestrate the gentle pulsing and contracting needed for survival. Within these networks, neurons developed early forms of synaptic communication. Though far simpler than modern synapses, they carried the same basic principles described in your uploaded materials. Neurotransmitters crossing tiny spaces to influence neighboring cells, ions flowing across membranes, and impulses shaped by excitation and inhibition. The early synapses were likely chemical rather than electrical, relying on slow but reliable diffusion of signaling molecules. Over time, these chemical messages became more refined, allowing specific patterns of activation to guide distinct behaviors. These early networks also gave rise to a primitive form of sensory integration. Although the organisms lacked specialized organs, certain cells within their tissues became more sensitive to touch, light, or chemicals. These sensory cells sent signals into the surrounding nerve net, where the information mingled with other signals before spreading outward. This integration allowed the organism to respond not just to direct contact, but to broader environmental conditions. Light-sensitive cells could trigger contractions that oriented the organism toward or away from sunlight. Chemical-sensitive cells could guide it toward nutrient-rich water. In this way, even simple creatures developed a sense of the world beyond direct physical contact. Movement in these early organisms became increasingly refined through the interplay of sensory and motor responses. The nerve net could generate waves of contraction that rippled around the body, allowing the organism to change shape or propel itself more effectively. In anchored animals like ancient sea anemones, the net allowed them to bend, sway, and withdraw into protective postures. These movements, though slow, represented the first coordinated motor actions in the history of life, and they depended entirely on the distributed logic of the nerve net. Another quiet innovation of nerve nets was the emergence of basic reflexes. A reflex is a simple pattern of response that occurs automatically when a stimulus is detected. In early animals, reflexes took the form of immediate contractions or expansions. When something brushed against the organism, it could contract its entire body in an instant, pulling its delicate tissues inward. These reflexes required no decision, no central interpretation. They were built into the structure of the net itself, emerging from the direct connections between sensory and contractile cells. Though nerve nets lacked the directional flows that later animals would develop with centralized nervous systems, they provided something essential, a foundation. They proved that neurons could work together, that distributed networks could coordinate movement, and that communication pathways could evolve toward greater complexity. They showed that life could sense, integrate, and respond in organized ways, laying the groundwork for the evolution of more advanced nervous systems. As time passed, some organisms began to develop slight variations in their nerve nets. Certain regions became denser, forming primitive clusters of neurons where signals gathered and intersected more frequently. These clusters would eventually become early nerve rings or ganglia structures that hinted at the possibility of centralization. Many creatures continued to rely solely on diffuse networks, and even today, some still do. But in others, the gathering of neurons in particular regions marked the first step toward a head, a brain, and more complex behaviors. In the calm of these early oceans, Life was experimenting quietly with new designs. The nerve net, simple and fluid, served as one of nature's earliest attempts at creating an internal communication system. It was flexible, resilient, and sufficient for the needs of the organisms that carried it. It allowed them to move, to protect themselves, and to interact with the environment in ways far richer than anything single-celled organisms could achieve. As we rest in this understanding, it becomes clear that nerve nets were not merely a stage in the evolution of the nervous system. They were the first expression of coordinated life. They bridged the gap between individual cells and the complex neural architectures that would one day allow animals to navigate the world with intention and precision. In their quiet simplicity, they carried the earliest patterns of response that would eventually give rise to minds capable of reflection, memory, and wonder, a journey that continues now as we move toward the emergence of centralized neural structures in the next part of this gentle, unfolding story. 7. Bilateral symmetry and the first brain centers. In the slow unfolding of early animal life, after nerve nets had begun weaving their delicate networks through simple bodies, evolution drifted toward a new and quietly transformative idea, the idea of direction. For countless generations, many early organisms lived in radial symmetry, arranged like ripples or flowers, able to respond equally from any angle. But the world around them was changing. Light traveled from a particular direction. Currents flowed along specific paths. Food sources drifted with patterns that had a beginning and an end. Predators moved through the water with speed and intention. In such a world, those organisms that could orient themselves, those that could distinguish a front from a back, gained an advantage, and so began the rise of bilateral symmetry, a moment in evolutionary history that set the stage for the first centralized brain-like structures. Bilateral symmetry may seem simple from a modern perspective. Many animals today, worms, insects, fish, birds, mammals, exhibit this balanced arrangement along a central line. But its emergence was profound. Once an organism acquired a defined front end, that region naturally became the place where sensory structures would gather. The front met the environment first. It encountered nutrients, obstacles, threats, and opportunities. As a result, evolution began concentrating sensory cells near this forward region, and where sensory cells gathered, signaling cells followed. Neurons connected these sensory areas to parts of the body that could respond: muscles, contractile tissues, and other neural clusters. Gradually, the front end of the organism became a dense hub of communication, forming the earliest prototypes of brain centers. These early bilateral creatures lived in the oceans, gliding across soft sediments or swimming gently through filtered light. They were not complex in form. Many were small, flat-bodied organisms that could move with modest undulations. Yet the symmetry they possessed allowed them to streamline their behavior. Instead of reacting equally to stimuli from all directions, they could orient themselves with purpose. Light-sensitive patches on their front could detect gradients, guiding them toward favorable conditions. Chemical sensors could help them locate food by following trails of molecules drifting through the water. Mechanical receptors could detect vibrations, warning them of an approaching predator. These sensory inputs, simple individually, became powerful when integrated in a central cluster. This integration marked a shift from distributed networks, like nerve nets, to the beginnings of centralized processing. Whereas nerve nets allowed signals to travel in every direction without clear hierarchy, these new clusters gathered information, sorted it, and then shaped a coordinated response. This early form of centralization did not resemble the brains of modern animals, yet it carried the same basic principle described in your uploaded materials. Sensory input arrives, the information is integrated, and motor commands are sent out. Even at this foundational level, we see the earliest blueprint for what would eventually become the brain's essential function. The first brain centers, often referred to as cerebral ganglia in ancestral organisms, were small, dense knots of neurons located at the front end of the body. They were not highly specialized. Instead, they acted as multifunctional hubs where sensory signals converged. These signals might come from light-sensitive cells, chemical receptors, or simple mechanical detectors. The central cluster processed this information and then coordinated a response through the rest of the nervous system. For early bilateral animals, this allowed smoother and more effective movement. Instead of reacting piece by piece, the organism could initiate whole-body actions with deliberate direction. Movement became more purposeful in these creatures. With a defined front end, they could glide toward food or away from danger. Their bodies often became elongated, allowing more efficient locomotion. This elongation, in turn, encouraged the development of a longitudinal nerve cord running down the body's length. That cord carried signals between the front-end cluster and distant parts of the organism. This design was another early step toward the spinal cord seen in vertebrates and other lineages. In these ancient creatures, the nerve cord worked like a communication highway. It ensured that signals from the front reached the back quickly and consistently, allowing the entire body to act as a unified whole. The presence of a nerve cord also supported more complex reflexes. Simple reflex patterns had already appeared in nerve nets, but in bilateral animals, these reflexes could be modified or guided by the front-end cluster. The earliest form of top-down control, primitive though it was, began here. A stimulus detected near the head could initiate a coordinated cascade of movement through the rest of the organism's body. This level of integration, even in its simplest form, represented a significant step in the evolution of behavior. It allowed organisms to begin making distinctions between competing stimuli, prioritizing one over another, and reacting with slightly more nuance than before. These early brain centers were also influenced by the growing diversity of sensory cells. Light-detecting patches became more organized, forming the earliest structures that would one day resemble eyes. Chemical sensors became more sensitive, allowing creatures to navigate richer and more varied environments. Mechanical sensors evolved to detect subtle shifts in pressure or touch, offering early warnings of approaching threats. The clustering of these sensory systems was not accidental. With bilateral symmetry, the front end naturally encountered the world first, and the neural concentration in that region allowed the organism to interpret these signals efficiently. Though simple, this early centralization marked the beginning of cephalization, the evolutionary trend toward developing a head region with concentrated sensory and neural structures. Cephalization is one of the major steps in nervous system evolution. Once organisms began collecting neurons at their front end, evolution had a new structure to refine. Over time, these clusters could grow larger, add new layers of specialization, and link to more complex motor systems. The earliest brain centers thus became stepping stones toward the richly layered brains seen in fish, reptiles, birds, mammals. Another important part of this transition was the emergence of directional behavior. In nerve net organisms, movement was often a general contraction or expansion, but in bilateral organisms with early brain clusters, movement gained direction. The body had a front, a back, a left, and a right. This spatial structure allowed for more controlled navigation. Animals could follow gradients, pursue prey, avoid obstacles, or retreat from threats with increasing accuracy. Once direction became relevant, the nervous system began developing internal maps. Simple at first, but meaningful. Neurons in the front dealt with what lay ahead, and neurons farther down the body coordinated the appropriate responses. This separation of roles among neurons echoes the broader biological pattern noted in your uploaded texts. Specialized cells, forming organized systems, each with its own function, yet all contributing to the larger whole. The early brain centers fit naturally into this framework. They became the organizers of sensory input and motor output, guiding the organism in ways that earlier forms of life could not achieve. In many of these early creatures, The central clusters also began to interact with emerging patterns of behavior, linking sensory cues to stored responses. Though memory in these organisms was extremely rudimentary, this might take the form of a slightly quicker reaction to touch or a more refined orientation toward light. These simple experiences marked the beginning of associative responses, an important precursor to learning. As we reflect on this stage of evolution, it becomes clear that bilateral symmetry did more than reshape animal bodies. It reshaped the flow of information itself. It created a natural logic for organizing the nervous system. Sensory input arrives at the front, the central cluster processes it, and motor commands travel down the length of the body. This logic remains present in animals today, including humans, whose sensory organs cluster around the head and whose spinal cord carries signals throughout the body. The structures that support vision, hearing, smell, and taste all developed from the same quiet evolutionary pressures that once shaped those early bilateral creatures drifting in the ancient sea. In this gentle arc of time, the first brain centers arose not as conscious minds, not as thinking centers, but simply as hubs of coordination, small yet powerful beginnings. These hubs allowed life to move with intention, to interpret the world with increasing clarity, and to forge the first true pathways toward complex behavior. This slow emergence of centralization would continue to unfold, giving rise to segmented brains, more refined sensory organs, and eventually the rich neural landscapes that define the living world today. 8. The Cambrian expansion of sensory worlds. In the soft, drifting stillness of evolutionary time, long after the first brain-like clusters had begun guiding the movements of simple bilateral creatures, the world entered a period of profound transformation. This era, known today as the Cambrian period, unfolded more than 500 million years ago, at a time when life was still primarily confined to the oceans. Yet the changes that occurred then reshaped the course of evolution in ways that can still be felt in every modern animal, including humans. The Cambrian period brought an explosion of diversity, an expansion of sensory abilities, and an unprecedented growth in the complexity of nervous systems. It was in these ancient seas, filled with drifting organisms and shifting currents, that the senses of sight, touch, smell, and movement underwent their first true flourishing. And with these senses came the need for more organized neural processing, setting the stage for the next great steps in the evolution of the brain. To imagine the Cambrian seas is to picture a world shimmering with life in forms that had never been seen before. Creatures with shells, spines, tentacles, appendages, segmented bodies, and even early eyes. Many of these forms appeared within a relatively short geological window, a blossoming of innovation that reflected life's growing ability to sense and interact with its surroundings. The oceans were no longer inhabited solely by simple worms or jelly-like beings. Instead, they became home to animals capable of active hunting, agile movement, complex feeding strategies, and subtle environmental detection. These abilities required new ways of gathering information and coordinating action. And so the sensory organs and neural systems of early animals began to expand dramatically. One of the most significant developments of this era was the refinement of vision. Before the Cambrian period, light-sensitive cells existed, but their function was limited. They could detect the presence or absence of light, helping organisms orient themselves, but they did not form images. During the Cambrian, however, clusters of these cells became more organized and more sensitive. Some organisms evolved cup-shaped structures that allowed them to detect the direction of light. Others developed lenses simple at first, but powerful enough to focus light into clearer patterns. These early eyes gave animals a new way to understand their surroundings. For the first time, creatures could detect shapes, movement, and contrast, allowing them to distinguish prey from background, predator from shadow, and open water from obstructed space. With the rise of vision came a profound shift in ecological roles. Animals that could see gained new opportunities, and animals that could not had to evolve strategies to avoid detection. This visual interplay created an evolutionary feedback loop. Predators evolved sharper imaging capabilities to track their prey. Prey evolved better camouflage or faster escape behaviors. The nervous system became increasingly important as animals needed to interpret visual information rapidly and respond appropriately. What began as simple light detection had transformed into a dynamic interplay between sensory input and motor action, a cycle that required more neural sophistication. Early animals developed arrays of mechanoreceptors, cells sensitive to pressure, vibration, and stretching. These receptors allowed animals to sense disturbances in the water. Touch became an essential tool for survival, especially for creatures that lived in sediment or near the ocean bottom. Many animals evolved long filaments, antennae-like structures, or sensory hairs that extended outward, acting as delicate feelers that scanned the environment. These structures were often connected to increasingly dense neural pathways, reflecting the need for swift interpretation of tactile information. Chemical sensing underwent a similar expansion. Primitive smell and taste receptors became more specialized, allowing animals to detect and interpret chemical gradients across larger distances. This ability helped organisms follow trails of nutrients or recognize the presence of predators. Chemical signals, once subtle and diffuse, became guiding forces that influenced movement and behavior. Even in simple organisms, these chemical cues required integration by neural circuits, reinforcing the importance of centralized processing regions. Movement itself became more complex during the Cambrian period. As new body shapes evolved segmented forms, articulated limbs, fin-like appendages, animals needed more precise control over their actions. Some creatures developed undulating motions that allowed them to glide smoothly near the ocean floor. Others learned to burrow, swim, or crawl with newfound agility. These refined movements demanded neural circuits that could coordinate multiple muscle groups in synchronized patterns. The early nerve cords present in bilateral organisms began to branch, forming segmental ganglia that managed localized movement while still communicating with the central cluster. The nervous system was becoming not just a communication network, but a control system capable of producing fluid, adaptive motion. In addition to movement and sensing, neural integration became increasingly sophisticated. Early animals needed to combine information from multiple senses, sight, touch, chemical detection, and produce rapid responses. This integration required new organizational structures in the nervous system. Some regions of the brain-like clusters began to specialize, handling specific types of sensory input. Though these specializations were simple compared to the intricate brain regions of later animals, they marked the beginning of modularity in the nervous system. Life was learning to separate functions into different neural domains, creating efficiency and clarity in processing. Another important development during the Cambrian period was the emergence of more advanced reflexes and patterns of behavior. Reflexes became faster and more tailored to specific environmental cues. Some organisms developed escape responses triggered by sudden changes in light or movement. Others evolved feeding reflexes that activated when prey came near. With these behaviors came the earliest glimmers of internal states, patterns of neural activation that represented hunger, readiness to flee, or exploratory curiosity. These states were not conscious, but they were functional, guiding behavior in ways that enhanced survival. Neural plasticity also began to deepen during this era. Repeated stimuli could alter the strength of connections between neurons, shaping the likelihood of certain responses. This capacity allowed organisms to adapt their behavior based on experience, a precursor to learning. Though extremely simple, this adaptability gave animals flexibility in environments that were increasingly diverse and dynamic. Your uploaded materials note the importance of the relationship between structure and function in biology, a principle that becomes vividly clear during the Cambrian. The sensory structures that evolved eyes, antennae, mechanoreceptors were matched by neural structures capable of supporting their functions. As sensory capabilities expanded, the nervous system expanded with them, forming circuits that allowed animals to navigate, pursue, and evade with growing precision. As we rest in this part of the story, it becomes evident that the Cambrian expansion was not just a flourishing of life forms, but a flourishing of sensory worlds. Animals went from dimly sensing their environments to perceiving them with clarity and depth. Sight sharpened, touch diversified, chemical detection grew more nuanced, and with each advancement, the nervous system adapted, deepening its complexity and organizing itself into the earliest forms of recognizable brain regions. The Cambrian seas shimmered with new possibilities. Each new sense opened new pathways for interaction, and each new interaction offered new opportunities for evolution. In this gentle, ancient light, the building blocks of perception were laid down, forming the foundations that would eventually lead to the intricate sensory experiences of modern animals. And from these foundations, the evolution of the brain moved steadily forward, guiding life toward ever greater complexity in the ages to come. 9 segmented brains and the rise of early invertebrates. Evolution found itself exploring yet another gentle and transformative direction. The earliest organisms with bilateral symmetry had already begun to gather neurons toward their front end, forming small processing clusters that acted as prototypes of the brain. But as body plans grew more complex, especially in animals whose bodies were divided into repeating segments, nature discovered an elegant solution to a growing challenge. Each segment needed its own local control, yet each needed to remain in harmony with the whole. This quiet challenge gave rise to one of evolution's most influential innovations: the segmented nervous system. In the calm shimmer of early oceans, many creatures were beginning to organize their bodies into segments. These segments were not just repeated portions of anatomy, they were functional units. Each had its own muscles, sensory structures, and responsibilities. To coordinate such a body efficiently, evolution favored the development of clusters of neurons in each segment, clusters that could handle local movement and sensation. These early neural clusters, known as segmental ganglia, acted like small processing centers. They received signals from the surrounding region and generated immediate responses without needing to send every message all the way to the main neural center at the front of the body. This arrangement allowed for remarkable improvements in flexibility and control. Instead of relying on a single neural cluster to manage the entire organism, these early invertebrates could handle multiple tasks simultaneously. A disturbance near the middle of the body could trigger a localized reflex, while the front of the animal continued guiding overall movement. The segmentation created a kind of distributed intelligence, a system in which each part understood its role yet remained deeply connected to the whole. The best-known examples of these early segmented organisms appeared among the ancestors of modern arthropods, annelid worms, and various other invertebrates. Their bodies were divided into rings or plates, each with its own repeating pattern of muscles and sensory structures. These organisms did not simply grow longer, they grew more structured, and as structure increased, so did the need for neural coordination. Segmental ganglia evolved to meet this need with quiet precision. Each ganglion served as a local command center. It processed information from sensory cells in its segment, such as vibrations in the surrounding water or contact with the seafloor. It interpreted this information and sent corresponding signals to the local muscles, allowing each segment to adjust its movement accordingly. When many segments acted in sequence, their combined motions produced graceful waves along the body, a kind of synchronized dance that allowed creatures to swim, crawl, or burrow with remarkable efficiency. These waves of movement, guided by chains of ganglia, were among the first examples of coordinated locomotion controlled by a distributed nerve cord. In annelid worms, for instance, movement took the form of slow peristaltic waves, one segment contracting while the next relaxed, creating a gentle ripple that carried the organism forward. In early arthropods, the segments supported jointed limbs that moved in coordinated patterns, guided by neural circuits capable of controlling alternating motions. These patterns would later become the foundations of walking, climbing, swimming, and even flying. The presence of multiple ganglia also meant that early invertebrates possessed a kind of layered control. Sensory signals traveled upward through the nerve cord toward the central cluster, but they could also be addressed locally without needing centralized oversight. This reduced the time it took to respond to threats or opportunities. Quick reflexes became possible. An unexpected touch might trigger a sudden bend, while a shift in chemical gradients could cause an immediate change in direction. This arrangement echoed a principle described in your uploaded materials. As biological systems become more complex, they often develop layers of organization, with each level contributing to the overall function. The segmented nervous system was a clear expression of this principle. Each level, each ganglion handled its part of the process while remaining integrated into the larger whole. As evolution continued its quiet work, some of these ganglia grew larger and more specialized. The frontmost ganglia, located near the head region, because these ganglia received the bulk of the sensory information, especially from newly developed eyes, antennae, and other structures, they became more complex than their counterparts in the rest of the body. Over time, these front ganglia fused together into larger, more organized clusters. These clusters formed the first true brains among early invertebrates, capable of more intricate processing and coordination. The early brain of an invertebrate was typically divided into several regions, each responsible for handling different types of sensory information. Some regions processed light and movement, others interpreted chemical signals or mechanical vibrations. Still others coordinated complex feeding behaviors. Although these brains were small by modern standards, they represented an extraordinary step forward. Instead of simply reacting to stimuli, these animals could integrate multiple sources of information, compare them, and choose among different behavioral responses. This integration opened the door to new forms of behavior. Early invertebrates began to explore, forage, and navigate with increasing sophistication. Instead of drifting passively through the environment, they moved intentionally, guided by neural circuits that blended sensory input with stored patterns of response. The segmented architecture allowed them to adapt their movement to different conditions. They could speed up or slow down, change direction, or explore textures and surfaces with appendages designed for touch and probing. The evolution of segmentation also supported the emergence of more advanced sensory structures. In arthropods, for example, antennae became intricate tools of exploration. They could detect chemicals, vibrations, and even subtle air currents. The ganglia associated with these structures grew larger and more specialized, reflecting the need to process this rich sensory information. Meanwhile, visual organs became more complex, and the neural circuits associated with vision expanded accordingly. In the midst of these developments, the nerve cord itself grew more sophisticated. Instead of simply serving as a conduit for signals, it became a dynamic processing structure organized into two main components: lateral nerve cords and segmental ganglia. Each segment contributed to a larger network that could coordinate the animal's entire body. This neural architecture created a balance between central control and local autonomy, a design so effective that many modern invertebrates still use it today. The segmented nervous system also laid foundational patterns for later evolution. It provided a template for the differentiated nervous systems of vertebrates, in which local reflex centers exist within the spinal cord, even as the brain oversees higher-level decision-making. The simple logic of segmentation local control, supported by central guidance, became one of evolution's enduring strategies. In reflecting on this part of the story, we can appreciate how segmentation added a new layer of organization to the nervous system. Nerve nets had allowed for diffuse responses, and early brain centers allowed for centralized sensing and coordination. But segmentation created something in between: a distributed network that could act quickly, flexibly, and with remarkable precision. This hybrid design gave early invertebrates the ability to adapt to their environment in ways that earlier organisms could not. As we drift gently through this understanding, it becomes clear that the rise of segmented brains represented a quiet flowering of complexity. It allowed animals to move with purpose, sense with refinement, and respond with coordination. These early invertebrates did not possess thoughts or intentions as we understand them, but they held within them the beginnings of a system that would, over millions of years. With segmentation came the first intricate neural patterns, and from these patterns, the next steps in brain evolution emerged, guiding life toward even greater depths of complexity in the chapters ahead. 10. The vertebrate blueprint emerges. In the long, gentle drift of evolutionary time, after segmented invertebrates had filled the oceans with bodies guided by chains of ganglia and simple brains, nature began to explore yet another quiet and profound transformation. This shift would give rise to a lineage whose nervous systems were organized around a different kind of structure, one that brought both stability and remarkable potential for complexity. This was the emergence of the vertebrate blueprint, the foundational design upon which all fish, amphibians, reptiles, birds, and mammals, including humans, would eventually build. To understand this transition is to step deeper into the evolutionary story of the brain, moving from distributed networks toward a more centralized, protected, and intricately layered nervous system. The earliest vertebrates were not the scaled, bony fish familiar to us today. Instead, they were soft-bodied, eel-like creatures living hundreds of millions of years ago in ancient seas. These early forms, often compared to modern lancelets or hagfish-like organisms, possessed simple notochords, flexible rods running along the length of their bodies. While not yet true backbones, these supportive structures gave the body a kind of firmness that earlier animals lacked. This internal support allowed for smoother, more controlled movement, and it also created a natural axis along which a centralized nervous system could develop. Along this notochord, evolution formed a hollow dorsal nerve cord, a structure that would one day become the spinal cord. This dorsal positioning was a departure from the ventral nerve cord seen in many invertebrates. By placing the nerve cord along the back, evolution provided a protected pathway for neural communication. Over time, the dorsal nerve cord thickened and became segmented, forming roots that extended outward to muscles on either side of the body. These neural extensions allowed for more refined control over movement, supporting the gentle undulations and coordinated swimming motions that early vertebrates relied upon. At the front end of the dorsal nerve cord, the first true vertebrate brains began to appear. These early brains were small and simple, yet they already displayed the three-part organization that remains visible in all vertebrate species today: the forebrain, midbrain, and hindbrain. Each region handled different aspects of sensory processing and coordination, forming a basic layout that would later expand into the highly specialized structures described in your uploaded materials. Even at this early stage, the hindbrain coordinated balance and movement, the midbrain processed visual and auditory signals, and the forebrain began to handle chemical sensing and primitive forms of integration. The emergence of paired sensory structures was another defining characteristic of these early vertebrates. Eyes became more complex, benefiting from the protective placement near the front of the body. These early vertebrate eyes were capable of forming clearer images than most invertebrate eyes of the time, giving them an advantage in detecting predators or prey. The neural pathways associated with vision grew thicker, and specialized clusters of neurons formed to process this visual input. Taste and smell receptors also grouped into distinct sensory organs, creating more efficient and focused chemical detection systems. As the sensory organs grew more sophisticated, the brain structures associated with them expanded as well. For example, the olfactory bulbs, small clusters of neural tissue responsible for processing smell, formed at the front of the forebrain, marking one of the earliest examples of a function-specific brain region. This early specialization allowed vertebrates to navigate complex chemical landscapes in the ocean, following trails of nutrients or avoiding harmful substances. Movement became more fluid and coordinated in these early vertebrates due to the interplay of the spinal cord, segmental nerves, and the brain's developing motor regions. The spinal cord provided a direct communication channel between the brain and the rest of the body, allowing for faster and more organized movement patterns. Local reflex circuits within the spinal cord handled many routine motions automatically, freeing the brain from managing every detail. This arrangement, local reflexes supported by centralized oversight, resembles the layered organization seen in modern vertebrate nervous systems, where the spinal cord interacts constantly with the brain to produce smooth, adaptive movements. One of the quiet yet critical advancements during this stage was the evolution of the neural crest, a unique group of embryonic cells found only in vertebrates and their closest relatives. These cells migrated throughout the developing body, giving rise to sensory neurons, pigment cells, cartilage structures, and parts of the skull. The neural crest allowed vertebrates to evolve sophisticated sensory organs and protective features, laying the foundation for future anatomical complexity. Without the neural crest, the elaborate cranial structures needed to house and protect the brain would not have been possible. The evolution of a protective skull, whether cartilaginous or bony, was another major milestone. Encasing the brain within a protective structure reduced the risk of injury, which meant that brains could grow larger without jeopardizing survival. This protection allowed natural selection to favor increased neural development, leading to more refined sensory processing, improved motor coordination, and the possibility of learning. The skull also provided stable attachment points for jaw structures, which would later influence feeding strategies and further promote nervous system development. As vertebrates diversified, different lineages began to emphasize different sensory modalities. Some developed larger eyes and more elaborate visual regions in the brain, allowing them to navigate clearer waters or pursue swift prey. Others expanded their olfactory regions, relying heavily on chemical cues. Still others refined their balance organs, using the inner ear to sense movement and orientation with exceptional accuracy. Each of these sensory advances required corresponding improvements in neural processing, pushing the brain to grow more intricate networks and more specialized regions. The organization of the brain into paired hemispheres began during this time as well. Though the early vertebrate brain was still simple, the forebrain started to split into left and right halves, each connected by bundles of nerve fibers. This arrangement would later support redundancy, improved processing efficiency, and the potential for hemispheric specialization seen in more advanced animals. These neural advancements were complemented by changes in the body's other systems, all working together to support more active lifestyles. According to the principles noted in your uploaded materials, biological systems evolve in coordination. As vertebrates gained more refined nervous systems, their circulatory and respiratory systems evolved to provide the metabolic energy needed to support active movement. Muscular systems became stronger and more complex. Sensory organs became more precise. These developments formed an integrated whole, a unified vertebrate design that would prove incredibly adaptable. Reflecting on this period, one can see how the vertebrate blueprint represented a quiet leap forward in the evolution of the brain. The dorsal nerve cord offered a secure and efficient communication pathway. The three-part brain established the groundwork for future specialization. The emergence of paired sensory organs provided clarity and direction, and the development of a bony or cartilaginous skull allowed the brain to expand safely. These early vertebrates were not fast thinkers or complex problem solvers, but they carried within them the seeds of future intelligence. They possessed nervous systems organized in ways that would make further development possible. In the gentle currents of ancient oceans, these creatures swam with bodies guided by a protected, centralized nervous system, a system that was becoming more capable with each generation. This blueprint would carry forward into the great diversification of fish, onto land with the first amphibians, and eventually into the minds of birds and mammals. As this story flows onward, the vertebrate brain will continue its quiet expansion, shaped by new environments, new challenges, and new opportunities. The journey toward the modern brain was only just beginning, but the essential foundations had been laid, deep in the waters of Earth's distant past. 11 ancient fish and the evolution of movement. In the gentle sweep of deep evolutionary time, Once the earliest vertebrates had established a basic blueprint for centralized nervous systems, a new chapter unfolded beneath the calm surfaces of ancient seas. Life was becoming more dynamic, more exploratory, and more attuned to the shifting patterns of light, current, and opportunity. Among the earliest creatures to take full advantage of this evolving design were the ancient fish, soft-bodied at first, then gradually developing fins, stronger muscles, and more defined skeletons. With each subtle change, the nervous system adapted in turn, refining pathways that allowed these animals to glide, dart, hover, and navigate in ways far more sophisticated than the gentle drifting that had characterized life before. Movement is one of nature's oldest and most expressive forms of communication. Even before brains existed, life responded to currents and stimuli with contractions, expansions, and drifting motions. But the moment early vertebrates acquired stronger muscles anchored to internal supports, movement took on a new quality. It became purposeful, it became directional, and it became a driving force in the evolution of the brain. Ancient fish, living hundreds of millions of years ago, were among the first animals to fully explore the potential of coordinated, rhythmic movement across the spine. Their bodies, structured with repeating blocks of muscle known as myomeres, could produce sweeping waves that carried them through water with fluid grace. These waves of motion were controlled not only by the muscles themselves, but by the nerve circuits that ran along the length of the spinal cord. Each segment of the spinal cord housed simple neural patterns capable of producing rhythmic contractions. These patterns, known as central pattern generators, were early examples of neural circuits that could produce repetitive actions without constant input from the brain. In many ways, the spinal cord of ancient fish became a quiet partner to the brain, handling the details of movement while the brain focused on sensing the environment and guiding overall direction. This division of labor was an important evolutionary step. The brain began to function more as a coordinator than as a micromanager. Instead of controlling every contraction directly, it sent broad signals: start, stop, slow down, speed up, turn, and the spinal cord carried out the instructions. This architecture remains in modern vertebrates, including humans, where many reflexes and movement patterns originate not from the brain, but from spinal circuits that quietly maintain rhythms of walking, balance, and posture. As ancient fish explored their environments, their sensory systems continued to grow more refined. Vision became clearer, aided by lens structures that could focus light more precisely. This improvement in vision required expansions in the midbrain, especially in regions responsible for processing visual input. A clearer view of the environment meant that fish could track moving objects. The neural pathways carrying this information grew stronger, and the brain regions interpreting it became denser with interconnected neurons. Touch also played an important role in aquatic environments. Early fish developed lateral line systems, rows of sensory receptors running along their sides. These receptors could detect vibrations and changes in water pressure, giving fish a sense of movement occurring anywhere around them. With this ability, ancient fish could locate prey hidden in murky water, detect the wake of approaching predators, and navigate through tight spaces with surprising sensitivity. The lateral line system acted almost like a secondary form of vision, one that worked even in complete darkness. And like all sensory structures, it required neural processing centers to interpret the incoming signals and produce coordinated responses. Smell and taste also expanded during this time. Water carries dissolved chemicals in delicate gradients, and the early fish learned to detect these gradients with increasing nuance. Chemical cues could signal food sources, suitable breeding grounds, or dangerous territory. As chemical detection became more important, the forebrain regions responsible for processing these signals grew. In many early fish, the olfactory bulbs responsible for interpreting smell became some of the largest structures in the brain, reflecting the importance of chemical communication in underwater environments. Another quiet but important development during this era was the refinement of balance and orientation. Swimming requires constant adjustment, and fish evolved inner ear structures that could detect changes in position, acceleration, and movement. These structures, which would eventually give rise to the vestibular system found in all vertebrates, connected to the hindbrain, allowing fish to maintain equilibrium even in complex currents. The nervous system learned to blend sensory information from vision, the lateral line, and the inner ear to produce graceful, controlled movement through three-dimensional space. As ancient fish grew more adept at navigating their environments, their bodies also began to change shape. Fins became more specialized, some for steering, some for stability, and others for rapid propulsion. Each refinement in fin structure required corresponding refinements in neural control. Muscles became more differentiated. These changes placed new demands on the brain, encouraging the development of more complex motor planning regions. The cerebellum, a structure deeply involved in coordination and balance, first began to expand significantly during this period. Even in early fish, the cerebellum served as a center for integrating sensory information with movement. It learned to predict the consequences of actions, adjusting motion patterns in real time based on feedback from the body and environment. This predictive capacity became one of the cerebellum's defining features and remains central to its function in modern vertebrates, supporting everything from swimming in fish to walking, speaking, and playing instruments in humans. The emergence of jaws in some early fish lineages further altered the landscape of neural development. Jaws allowed for new feeding strategies, biting, grasping, and manipulating food, and each of these behaviors required specialized motor circuits. The brain regions responsible for controlling jaw movement expanded, and the sensory systems supporting feeding behaviors grew more refined. The rise of jaws also encouraged more active predation, which, in turn, increased the demands on vision, coordination, and rapid decision-making. In this period, the beginnings of more advanced behavioral patterns also appeared. Ancient fish learned to navigate territories, follow seasonal migrations, and make use of protective structures in their environment. Though their behaviors were still largely guided by instinct and reflex, the neural circuits that supported them grew in sophistication. Sensory information from multiple sources was integrated into simple but effective internal maps, representations of where food, shelter, and mating opportunities could be found. The spine's role in this growing complexity cannot be overstated. The spinal cord served as a central conduit for information traveling between the brain and the body, but it also housed many of the fundamental circuits that made coordinated movement possible. The logic of this system, described broadly in your uploaded materials as the interaction between sensory input, integration, and motor output, became more structured and more efficient with each generation. Reflecting on this chapter of evolution, we can see how ancient fish laid many of the foundations that would support future vertebrate diversity. Their nervous systems grew not just in size, but in organization. Their sensory worlds expanded. Their bodies learned new ways of moving through their environment, and their brains learned new ways of guiding those movements. Every undulation, every flick of a fin, every shift in current became part of a larger dialogue between body and environment, a dialogue mediated by an increasingly capable nervous system. In these ancient waters, the vertebrate brain gained new dimensions of communication. These early innovations set the stage for life to leave the seas, to breathe air, to walk on land, and to explore worlds that had never before known vertebrate movement. And, as the story continues, the nervous system will evolve once again, adapting to new challenges, new environments, and new forms of life that would reshape the future of Earth's living history. 12. Leaving the water. Amphibians and adaptation. In the slow and ancient rhythm of evolution, after the early fish had refined the patterns of movement and sensory coordination that let them glide gracefully through the oceans, a quiet turning point began to unfold. The earth itself was changing. Shorelines expanded, wetlands formed, plants began to reach above the water's surface, creating shaded pools and tangled roots that offered both opportunity and challenge. And within these shifting landscapes, some early vertebrates started to explore the delicate boundary between water and land. Their bodies were still shaped by the sea, yet their nervous systems were beginning to sense the possibilities of a new world filled with unfamiliar sights, sounds, and textures. This gentle transition from water onto land marks one of the most profound evolutionary steps in the history of the brain. To leave the water, even partially, required a host of new abilities. Movement, vision, hearing, balance, and breath all had to be reimagined in subtle ways. The underwater environment had supported fish with buoyancy that made motion smooth and effortless, but on land, gravity pulled directly on their bodies, demanding stronger limbs and more precise neural control. Early amphibian-like creatures, descendants of lobe-finned fish, began developing bones, muscles, and joints that could support their weight. Their fin rays thickened, forming the precursors of digits, and their shoulders and hips became more robust. Each anatomical change required the nervous system to adapt, creating new circuits for coordination on uneven surfaces. These early movements on land were slow and tentative. Creatures such as the ancient Tiktaalik are believed to have spent much of their time in shallow water, lifting themselves briefly onto mudflats to rest, bask, or escape predators. With each small venture onto land, their brains gathered new sensory information, dry air brushing against moist skin, the warmth of sunlight, the subtle vibrations of the ground beneath them. These sensations were processed by expanding neural systems that had to integrate both waterborne and airborne cues. The brainstem, responsible for regulating basic functions, began adapting to manage breathing patterns that shifted between gills and primitive lungs. This interplay between water and air demanded flexible control, and evolution answered with expanded neural regulation. Vision changed as well. Underwater, light behaves differently. It scatters, softens, and loses clarity. On land, light carries sharp contrasts, deep shadows, and vivid patterns. The eyes of early amphibian ancestors needed to adjust to this new optical environment. Lids and stronger muscles around the eyes offered protection and better focus, while neural circuits in the midbrain adapted to process clearer, more intense visual input. The world above the surface was full of new shapes and movements, and the nervous system expanded to interpret these cues with increasing accuracy. Hearing evolved in gentle but significant ways. In water, sound travels rapidly and envelops the listener, but in air, sound is slower and more directional. Early amphibians developed simple tympanic structures, precursors to eardrums that allowed them to detect airborne vibrations. These vibrations traveled to small bones within the head, eventually forming the earliest versions of the middle ear. The brain's auditory centers began to differentiate, learning to interpret these new types of signals. This marked one of the earliest steps toward the complex hearing abilities found in later vertebrates. Balance and orientation also had to adjust. Underwater, ancient fish relied heavily on the lateral line system to detect movement and pressure changes. But on land, that system lost much of its usefulness. Instead, the vestibular system within the inner ear became increasingly important. Tiny, fluid-filled chambers with delicate hair cells helped these early amphibian forms sense tilt, acceleration, and orientation relative to gravity. Neural pathways connecting the vestibular system to the cerebellum grew stronger, supporting the careful and coordinated limb placement needed to move across rocks, mud, and vegetation. As these early creatures began to spend more time on land, their respiratory systems deepened in complexity. They relied less on water flowing through gills and more on drawing air into lungs. This shift required new patterns of neural control, because breathing in air is more rhythmic and voluntary than the passive flow of water across gills. The brainstem developed circuits that timed inhalation and exhalation, regulated airflow, and adjusted breathing in response to changing oxygen levels. This rhythmic breathing, regulated by the nervous system, became one of the fundamental patterns of vertebrate life, a pattern so deeply embedded that it remains part of the quiet rhythm that guides our own rest and sleep. The neural circuits controlling movement also grew more refined. Walking or crawling on land demanded precise coordination between limbs, which required the spinal cord to integrate signals differently than when controlling swimming. Muscles needed to contract in alternating pairs, and the timing of these contractions had to adapt to varied terrain. The brain communicated broad intentions: move forward, climb, turn, and the spinal cord worked out the details. This division of labor, first refined in ancient fish, became more elaborate in early amphibians, supporting a richer repertoire of movement patterns. These early excursions onto land also changed the way animals interacted with their environment. Smells behaved differently in air, spreading widely and lingering in still spaces. The olfactory system of early amphibians expanded, and the forebrain regions associated with smell grew correspondingly. Smell became one of the primary senses guiding behavior on land, helping animals find food, identify other members of their species, and avoid danger. Touch became more important too. Skin that had once been surrounded by supportive water now made direct contact with diverse surfaces: soft mud, coarse sand, rough stone. Nerve endings in the skin transmitted this new sensory information to the spinal cord and brain, helping the animal navigate more confidently. This heightened tactile sensitivity played a crucial role in shaping behavior and refining motor control. The earliest amphibian-like creatures needed to balance time in water with time on land. They returned to water to lay eggs and to stay moist, yet they gained access to new feeding opportunities on land. This dual lifestyle demanded a flexible nervous system that could guide behavior across two very different environments. Simple learning patterns began to emerge: associations between certain smells, textures, or temperatures, and the behaviors needed to survive. These neural patterns echoed the principles highlighted in your uploaded materials, and the constant flow of sensory input guiding motor output. Each sensory expansion shaped the nervous system, and each new neural adaptation made further sensory exploration possible. In time, as early amphibians grew more comfortable on land, their brains continued to expand. The midbrain refined its visual and auditory processing pathways. The hindbrain strengthened its control over movement and balance. These changes were subtle across generations, yet they represented a profound shift in how vertebrates interacted with the world. As we reflect softly on this moment in evolutionary history, we can see how the nervous system was shaped not by sudden leaps, but by countless small adaptations. Each step onto land offered new information, new challenges, and new opportunities for neural growth. The creatures that first bridged the gap between water and land carried within them the earliest versions of the neural circuits that would one day support climbing birds, running mammals, and eventually humans walking upright on open plains. In this long and gentle transition, the vertebrate brain deepened its capacity for coordination, sensory integration, and environmental awareness. The world above the water introduced new dimensions to life, and the nervous system rose to meet them with quiet ingenuity, laying the groundwork for the next stages of evolution on land. 13 reptiles and the foundations of the limbic system. In the unfolding calm of evolutionary history, after the earliest amphibians had taken their tentative steps onto land and begun exploring the dual worlds of water and air, nature continued its patient work. The land masses of Earth grew warmer and drier, the swamps receded, and new challenges emerged that demanded fresh adaptations. From these shifting environments arose the early reptiles, creatures that would change the course of vertebrate evolution by mastering life fully on land. Their bodies, behaviors, and nervous systems matured into forms that could thrive in the open sun, withstand long dry seasons, and explore vast territories. And within their developing brains, something subtle yet profoundly influential began to bloom: the earliest foundations of what humans would one day call the limbic system. Reptiles represented a departure from the moist dependency of amphibians. No longer tied to water for reproduction, they evolved amniotic eggs with protective shells and membranes, allowing their young to develop in secure, self-contained environments. This independence opened new ecological spaces, and with each step into these new worlds, reptiles refined their sensory systems, their motor patterns, and their neural circuitry in ways that ripple through the biology of all later vertebrates. One of the most important evolutionary changes in reptiles was the enhancement of their sensory perception. The world they inhabited was bright, open, and filled with new cues. Reptilian eyes grew sharper, more capable of detecting motion at a distance and discerning shapes against richly lit backgrounds. The visual centers within the midbrain expanded to handle this increased flow of information. For animals living under the intense sun, sight became the primary guide for hunting, avoiding predators, and navigating wide territories. Hearing also improved. While amphibians had only started to detect airborne vibrations, reptiles refined this sense. Their ears developed more efficient structures for capturing sound, and the bones within their heads transmitted vibrations with greater clarity. These improvements required neural adaptations as well, encouraging the expansion of auditory pathways and deeper integration between sound and action. Touch remained important, but its role began to shift. Reptilian skin, covered in scales, provided protection from the harsh environment but also limited direct tactile sensitivity. However, specialized receptors still existed beneath the scales, allowing reptiles to sense pressure changes, ground vibrations, and subtle shifts in temperature. These tactile cues helped them sense approaching animals, find shelter, and regulate body temperature. Smell, one of the earliest senses in vertebrate evolution, became exceptionally refined in many reptile lineages. Chemical detection allowed reptiles to interpret environmental cues that sight or sound could not reveal. Some reptiles, such as snakes, developed highly specialized systems, including the Jacobson's organ, their ability to detect chemical trails. This expansion of olfactory ability was mirrored by a corresponding growth in the brain regions, responsible for processing smell regions that would later contribute to the limbic system. As their sensory worlds expanded, reptiles developed new patterns of behavior that required more intricate neural control. They became true masters of terrestrial locomotion, moving with confidence across sand, rock, and forest undergrowth. Their muscles and limbs adapted for walking, running, climbing, or burrowing. These diverse movement patterns demanded precise coordination between the hindbrain, spinal cord, and cerebellum. The cerebellum, already growing in ancient fish and amphibians, became even more critical in reptiles, fine-tuning balance and timing so that movements could be quick, efficient, and controlled. But it was not only physical movement that deepened during this era. Reptiles also developed new internal states, patterns of behavior guided by hunger, territoriality, reproduction, and survival. These internal impulses required neural structures capable of integrating sensory input with stored instincts and past experience. It is here that the earliest foundations of the limbic system began to take shape. The limbic system, as described in part within your uploaded materials, is associated with emotion, memory, and instinctive behavior. Its early forms did not generate emotions in the human sense, but they did provide reptiles with layers of motivational behavior far more complex than those of amphibians. Key structures such as the amygdala and hippocampal precursors began to emerge in these ancient brains. The amygdala contributed to survival behaviors like fear responses, aggression, and territoriality. A sudden movement or unfamiliar scent could trigger defensive postures or rapid escape. The organism was beginning to recognize not just the immediate sensory world, but meanings within that world, signals of danger, opportunities for food, or chances to reproduce. These patterns created early emotional tones, instinctive but deeply influential, guiding reptiles in making quick decisions that increased their chances of survival. The hippocampal regions, developing alongside the amygdala, supported spatial memory. Reptiles needed to remember where water sources were located, where they had encountered prey, and which areas provided shelter from predators or harsh temperatures. This spatial mapping allowed reptiles to navigate more effectively across their territories. These early memory circuits offered a degree of continuity in behavior, enabling animals to respond based not only on current sensory input but also on past encounters. This small but powerful ability to learn from experience was one of the cornerstones of the limbic system's early evolution. Reptiles could adapt their strategies, avoid repeated mistakes, to what mammals would achieve, yet it marked a turning point. The nervous system was no longer responding only to immediate stimuli. It was beginning to weave time into behavior. As the forebrain grew to house these new structures, the midbrain and hindbrain continued to refine their functions. The reptilian brain still relied heavily on instinct and reflex, but the growing limbic components added flexibility. Behavior became less rigid. Decisions, though still instinctual, were informed by memory and context. The brain could weigh environmental cues differently depending on internal states, such as hunger or stress. These shifts foreshadowed the more complex emotional lives of later animals. Reptiles also began to develop social behaviors in some lineages, though still simple in form. Courting rituals, territorial displays, and parental guarding of eggs appeared sporadically, adding to the need for neural circuits capable of interpreting the behaviors of other members of their species. These interactions required the nervous system to integrate sensory cues from multiple sources, visual patterns, chemical signals, and movement cues into coherent meanings. Reflecting on this evolutionary stage, we see a deepening of internal processes that resembles the broader theme emphasized in your uploaded materials: the interaction of body systems to form a coordinated whole. The nervous system, endocrine system, and sensory structures evolved in parallel, shaping one another. Hormones influenced behavior, sensory cues guided hormonal cycles, and neural circuits mediated both systems. These interwoven mechanisms allowed reptiles to thrive in environments that demanded both resilience and adaptability. The journey from amphibians to reptiles was not a sudden leap, but a long series of small changes that accumulated quietly across generations. Each change deepened the relationship between sensory input, internal states, and behavior. Each adaptation refined the architecture of the brain, and with each subtle shift, the early limbic structures grew more influential, laying down patterns that would one day support the emotional landscapes of mammals and the rich tapestry of human experience. In this tranquil moment of understanding, we can appreciate how reptiles, with their alert eyes, patient stillness, and sudden bursts of motion, represent a pivotal point in the evolution of the brain. They carried within them the seeds of emotional processing, spatial memory, and instinctive behavior, features that would guide all later vertebrates as evolution continued to unfold across the warm, sunlit landscapes of Earth. 14 birds, and the high-speed brain. In the gentle continuum of evolutionary time, after reptiles had spread across the dry and sunlit landscapes of Earth, a new lineage began to emerge. One that would eventually fill the skies with motion, color, and song. The ancestors of birds were small, agile, and alert creatures, living in a world that demanded quick decisions and rapid coordination. From these early theropod dinosaurs, evolution began shaping bodies that were lighter, more balanced, and capable of extraordinary movement. And as these bodies changed, their nervous systems changed with them, refining circuits of perception, timing, and control that would eventually make birds some of the most neurologically remarkable animals on Earth. To understand the nervous system of birds, it helps to imagine the early forms that preceded them. These were creatures who needed to leap quickly, run swiftly, and maintain awareness of predators and potential prey at every moment. Their lives unfolded in bursts of action, darting from cover, climbing, gliding, or snapping at small insects. Such a lifestyle required a brain capable of processing information at great speed. The reptilian brain had laid the foundations for sensory integration and instinctive behavior. But the step toward true flight would require even greater refinement. Flight is a complex, delicate act, demanding perfect timing of wings, balance adjustments, rapid visual processing, and moment-to-moment coordination of many body systems. For flight to evolve, the brain needed to become faster, more efficient, and more specialized. As feathers evolved from scales, initially perhaps for insulation or display, and later refined for aerodynamic use, the nervous system began adapting to manage the new possibilities these structures offered. Wings, especially, introduced new challenges. They required muscles arranged in intricate layers and coordinated through precise neural signals. The spinal cord developed more specialized pathways to control the sweeping, rhythmic contractions of flight muscles. The hindbrain, already deeply involved in balance and movement, became even more critical. With every beat of the wings, sensory feedback flowed into the cerebellum, which adjusted timing and orientation in real time. The cerebellum in birds is proportionally enormous when compared to that of their reptilian ancestors. This expansion supports the constant stream of calculations that keep a bird's body steady in the air. Calculations involving direction, speed, wind resistance, and changes in altitude. Vision, too, became vastly more refined. Birds inherited the basic visual structures from reptiles, but their evolutionary path pushed these structures to new levels of clarity and speed. In the air, where distances stretch farther and light behaves differently, acute vision became essential. Many birds today possess some of the sharpest vision in the animal kingdom, capable of detecting small movements from great distances. This clarity required expansions in the midbrain and forebrain regions responsible for processing visual information. The optic tectum, already significant in reptiles, expanded even further in birds, becoming one of the primary centers for visual analysis. Here, signals arriving from the retina are interpreted with astonishing speed, allowing birds to correct their flight path instantly or capture prey midair. In addition to visual refinement, birds also developed more sensitive vestibular systems. The vestibular structures within the inner ear, which help animals understand orientation and balance, adapted to manage the complex motions of aerial movement. Sudden shifts, banking turns, gliding descents, and hovering all required real-time adjustments. Neural pathways connecting the vestibular system to the cerebellum and brainstem became highly specialized, enabling birds to maintain stability even during rapid maneuvers. While vision and balance grew increasingly elaborate, the sense of hearing underwent its own transformation. Birds rely heavily on sound for communication, for mate selection, and for recognizing members of their own species. Their auditory systems adapted to detect fine differences in pitch and rhythm. In species that developed song, such as early songbird ancestors, songbirds evolved nuclei devoted entirely to vocal learning and production, representing one of the earliest examples of culturally transmitted behavior among vertebrates. Neural circuits had to process, store, reproduce, and refine sound patterns, and this demand enriched the forebrain's complexity. The evolution of vocal communication added another layer to the expanding nervous system. Birds needed to remember songs, adjust their calls, and interpret the vocalizations of others. This required memory systems that were more flexible and capable of storing patterns with precision. The hippocampal regions earlier shaped in reptiles for spatial mapping grew in certain bird species to support memory-based navigation, especially in migratory birds that traveled vast distances. These regions also help birds remember food caches, territories, and nesting sites, further strengthening the connection between memory and survival. Flight also encouraged refinements in the limbic structures inherited from reptiles. Birds needed to respond rapidly to threats, protect their nests, and navigate social relationships within flocks. The amygdala-like regions in their brains became more complex, supporting a range of instinctive behaviors. Though birds do not experience emotion in the human sense, their limbic regions guide behaviors related to fear, bonding, territoriality, and courtship. These neural circuits shape their interactions, allowing them to establish stable social groups or defend essential resources. The forebrain of birds, once thought to be simplistic, is now understood to be deeply complex. It does not resemble the mammalian brain in shape, but its internal organization supports problem solving, tool use, memory, and learning in sophisticated ways. Corvids and parrots, descendants of ancient bird lineages, demonstrate remarkable intelligence. Studies of modern species reveal neural circuits that rival primates in certain tasks, circuits rooted in expansions that began millions of years ago as early birds refined their sensory and motor capacities. The high-speed lifestyle of birds also required efficient metabolic regulation. Their brains needed to operate with precision even when energy demands were high. This required close coordination between neural circuits and the endocrine system. Hormones guided migration, breeding cycles, and stress responses, while the nervous system interpreted environmental cues, day length, temperature, food abundance, and signaled the body to prepare for seasonal changes. This interplay reflects a theme found in your uploaded materials: the interconnectedness of body systems, each supporting the others in maintaining stability and function. The evolution of birds also brought new patterns of parental care. Many species began to incubate eggs, protect nests, and feed their young. These behaviors required complex neural circuits for recognition, memory, and coordination. Parental care often strengthened the bond between individuals, leading to the development of social communication systems more elaborate than those of most reptiles. Reflecting gently on this stage of the brain's evolution, it becomes clear that the rise of birds introduced a unique kind of neural precision to the living world. Their brains did not simply grow larger, they grew faster, more efficient, and more specialized. The demands of flight shaped pathways that could process information in fractions of a second. The needs of navigation encouraged memory systems capable of mapping continents. The songs of early species laid groundwork for communication patterns that would later echo in the complex vocalizations of mammals. In the quiet dawns of ancient forests, long before humans appeared, early birds took to the air with minds shaped by the balance of instinct, sensation, and rapid calculation. Their brains, small yet powerful, carried within them the beginnings of remarkable intelligence, a testament to the way movement, environment, and neural adaptation intertwine across time. As evolution continued to unfold, these high-speed minds would inspire new neural designs in mammals and eventually contribute to the foundation for cognitive complexity in the lineage that led to humankind. 15 early mammals and the expanding neocortex. In the soft, unhurried flow of evolutionary time, after birds had risen into the skies with swift minds and finely tuned senses, another lineage began its quiet ascent on the ground below. These were the early mammals, small, often nocturnal, and living in a world dominated by larger reptiles and early dinosaurs. While they did not yet possess the grandeur or complexity of later mammalian forms, these early creatures carried within their small bodies a set of profound innovations. Their nervous systems, shaped by new rhythms of life, began to expand and reorganize in ways that would eventually lead to the remarkable neocortical structures seen in humans today. In these early mammals, evolution planted the seeds for curiosity, learning, attachment, and the subtle emotional landscapes that distinguish mammalian experience. To imagine these early mammals is to picture gentle shapes moving quietly under ferns, navigating night forests with soft steps and careful awareness. Their survival depended not on size or strength, but on sensitivity, an ability to sense danger before it arrived, to remember safe hiding places, and to care for vulnerable young. This lifestyle placed unique demands on the nervous system. In the darkness, vision played a smaller role, while other senses, smell, hearing, and touch grew sharper and more refined. The expansion of these sensory abilities required the brain to process a greater diversity of information, and it responded by developing new structures and pathways that encouraged integration, memory, and flexible behavior. One of the defining features of mammals was the emergence of endothermy, the ability to maintain a stable internal body temperature. This shift required tremendous metabolic energy and closer coordination between body systems, including the nervous system, endocrine system, and circulatory system. As noted in your uploaded materials, biological systems work together continuously, exchanging signals in order to maintain balance and stability. For mammals, this coordination became even more essential. The brain needed to monitor temperature, adjust heart rate, and regulate metabolism with exquisite precision. This steady internal environment, in turn, allowed neurons to function more consistently, supporting the development of more complex cognitive processes. Another quiet transformation took place within the sensory systems. The mammalian ear evolved into a structure of remarkable sensitivity. Bones that once formed part of the jaw apparatus in reptiles became repurposed into delicate middle ear bones capable of transmitting sound with great clarity. This refinement gave mammals access to a rich world of sound, subtle rustling leaves, distant calls, and the quiet movements of nearby creatures. The auditory centers of the brain expanded to interpret these signals, and over time, mammals developed sophisticated communication systems based on tone, rhythm, and subtle vocal variations. Smell became equally important. Nocturnal environments rely heavily on chemical cues, and early mammals possessed large olfactory bulbs relative to their body size. These bulbs, part of the forebrain, processed information about predators, prey, mates, and territories. The olfactory pathways led deeply into the limbic regions, intertwining memory with scent and allowing mammals to retain lasting impressions of places and experiences. This strong connection between smell and memory remains a characteristic feature of mammalian brains, even today. As sensory pathways expanded, so too did the structures responsible for integrating them. The cerebral cortex, initially a thin and modest layer, began to grow in thickness and surface area. It formed grooves and folds that increased the available space for neurons, allowing more intricate connections and more sophisticated processing. This growing cortical layer represented the early form of what would eventually become the mammalian neocortex, the structure central to learning, problem-solving, and conscious perception. The neocortex allowed early mammals to form flexible strategies rather than relying solely on instinct. Instead of responding rigidly to stimuli, they could learn from experience, adapt their behaviors, and anticipate future events. A mammal that encountered a predator in one location could later avoid that area. One that discovered a reliable food source could remember the path back. These abilities required circuits capable of storing, organizing, and retrieving complex memories, supported by interactions between the hippocampus, the expanding neocortex, and the sensory systems. The limbic system, which had begun taking shape in reptiles, grew even more sophisticated in mammals. Emotional states, though still instinctive and not conscious in a human sense, became powerful guides for behavior. Fear sharpened attention. Curiosity encouraged exploration. Comfort and bonding strengthened social structures. The amygdala refined its ability to interpret emotionally relevant stimuli, while the hippocampal regions supported the creation of more detailed spatial maps and memory patterns. Perhaps one of the most profound developments in early mammals was the evolution of parental care. While reptiles often laid eggs and left them to develop alone, mammals invested tremendous energy into nurturing their young. This required strong social bonds, communication, and the ability to recognize and respond to the needs of offspring. The nervous system adapted to support these behaviors. Hormones such as oxytocin influenced bonding and maternal behaviors, while neural circuits within the limbic system guided nurturing actions. This emotional investment encouraged learning, cooperation, and the formation of early social structures, all of which demanded deeper neural integration. The motor systems of early mammals also evolved to support their active lifestyles. Their bodies became more agile, with limb structures capable of climbing, burrowing, or running with precision. The cerebellum, responsible for coordinating movement, expanded further to manage the balance and timing required for these tasks. Trunk and limb muscles needed fine control, as mammals often relied on stealth and agility rather than brute strength. Neural circuits in the spinal cord carried refined instructions from the brain, while sensory feedback from the environment continually adjusted movement in real time. The combination of heightened sensory awareness, emotional processing, and refined motor control gave mammals a uniquely flexible repertoire of behaviors. They could explore new environments, form social groups, care for their young, and learn from past experiences. These abilities marked a significant departure from the mostly instinct-driven behaviors of earlier vertebrates. The early neocortex, still small compared to later mammalian brains, supported these advances by integrating sensory input and guiding motor responses in more complex ways. Over millions of years, this structure would continue to grow, eventually giving rise to the extraordinary cognitive abilities of primates, cetaceans, and ultimately humans. But even in its early form, the neocortex represented a new horizon in the evolution of the brain, a structure capable of abstraction, foresight, and learning that extended beyond immediate survival. As we rest gently in the understanding of this evolutionary moment, we can appreciate how early mammals carried within them the beginnings of the emotional warmth, social bonds, and flexible intelligence that define their lineage. Their brains became more layered, more communicative. The night forests they navigated were alive with sound, scent, and sensation, and their nervous systems grew to interpret these cues with increasing clarity. In these quiet ancestors, the foundations of mammalian intelligence took root. Their expanding neocortex opened new pathways for behavior, memory, and adaptation, setting the stage for the remarkable diversity of mammals that would emerge in the ages to come. Creatures who would run across open plains, glide through treetops, swim through icy oceans, and eventually stand upright to contemplate their own minds. 16 primates and the architecture of social minds. In the long, slow unfolding of Earth's story, after early mammals had grown into agile, warm-blooded creatures with expanding neocortices and deepening emotional lives, the world continued to shift around them. Forests thickened and thinned through changing climates. Continents drifted. Within these branching possibilities, a particular lineage of mammals began to develop traits that would reshape not only their own destiny, but eventually the destiny of the entire planet. These were the early primates, creatures of grasping hands, forward-facing eyes, and increasingly intricate social lives. Their nervous systems deepened in complexity to meet the demands of climbing, exploring, communicating, and living among others. And within their developing brains emerged patterns of cognition, memory, and emotion that formed the early blueprint of the human mind. To picture the earliest primates, imagine warm forests, filtered with gentle light, filled with sprawling canopies and fragrant leaves. These environments encouraged a new kind of movement, one that required precise control, awareness of depth, and the ability to judge distances quickly. The ancestors of primates took to these trees with remarkable adaptability. Their hands evolved to grasp branches, their feet developed flexible digits, and their eyes rotated forward to better gauge space. This new visual alignment allowed for stereoscopic depth perception, one of the essential elements of primate life. With two overlapping fields of vision, the brain could calculate distances with greater accuracy, enabling the swift, fluid movements needed to leap from branch to branch. This shift in visual processing required expansions in the visual cortex, particularly in the occipital and parietal regions. The brain became more involved in interpreting shapes, shadows, and the subtle patterns of light through leaves. This visual complexity encouraged broader cortical development, strengthening networks that linked vision with touch, balance, and movement. Early primates relied on these connections constantly. A misjudged leap could mean a fall, and in the quiet but unforgiving world of the canopy, precision meant survival. With their sensitive hands, early primates also developed a new level of tactile awareness. The fingertips became rich with nerve endings, capable of detecting texture, pressure, and temperature with extraordinary clarity. This tactile world allowed them to explore their environment more intimately, feeling bark, leaves, fruit, and the presence of insects under the surface. The somatosensory cortex expanded to accommodate these new inputs, offering more detailed and nuanced representations of touch. Smell, while still important, became slightly less dominant as vision and touch expanded. But the olfactory regions of the brain remained essential for identifying food, recognizing group members, and interpreting subtle chemical signals in the environment. This balance between sensory systems began shaping a more versatile nervous system, one that could integrate inputs from multiple sources to create a cohesive understanding of the world. As early primates navigated the canopy, they also began forming social groups. Living in groups offered protection, allowed shared care of young, and increased access to resources. But it also introduced new challenges. Understanding social relationships required the nervous system to process facial expressions, vocal calls, posture, and emotional cues from other members of the group. This social complexity encouraged the growth of brain regions involved in communication, empathy, and memory. The limbic system, which had first expanded in reptiles and matured in mammals, became even more intricate in primates. Connections between the amygdala, hippocampus, and frontal regions strengthened, supporting emotional understanding and social learning. Within these early groups, the seeds of cooperative behavior took root. Grooming became a form of bonding, vocalizations became more nuanced, and young primates learned from observing older individuals. These interactions required the brain to track relationships, remember past encounters, and predict future behaviors. Neural circuits began forming around concepts of trust, hierarchy. Though simple in comparison to later primate and human societies, these early structures laid the foundation for social cognition, an ability essential to primate survival. The frontal lobes, particularly the prefrontal cortex, began to expand during this time. This region supports planning, decision-making, impulse control, and understanding cause and effect. Even in early forms, the prefrontal cortex allowed primates to evaluate situations more flexibly. They could weigh risks, choose alternate paths, and adapt their strategies when solving problems. A primate might plan how to reach fruit on a distant branch, or decide whether to join a group member in exploration. These abilities signaled a shift from instinct-driven behavior to more thoughtful action. At the same time, memory systems became richer. The hippocampus, responsible for forming and organizing memories, grew more elaborate, allowing primates to remember not only the layout of their environment, but subtle details about objects, food sources, and social interactions. This spatial and social memory made navigation safer and group life more meaningful. It also enabled a rudimentary sense of continuity and awareness that past events could influence present decisions. Communication among primates became increasingly important. Calls conveyed danger, location, or social intent, and body language expressed emotions like fear, curiosity, or affection. Neural pathways connecting auditory regions to the frontal cortex strengthened, allowing early primates to interpret vocalizations with greater nuance. This foundation would one day support the complex vocal communication seen in advanced primates and eventually in humans. Another subtle but profound evolution occurred in emotional processing. Mammals had already developed emotional states tied to survival and reproduction, but primates began experiencing emotions in ways that shaped their social behavior. Comfort, reassurance, anxiety, and playfulness all contributed to community structure. The brain adapted to support these states, reinforcing bonds and guiding interactions. As your uploaded materials emphasize, the nervous system works in harmony with other body systems, including the endocrine system, which influences mood, stress, and social bonding. Primates experience these hormonal and neural interactions in new and meaningful ways. Movement also became more refined. Primates relied on a combination of climbing, leaping, walking, and sometimes brief gliding motions. Their cerebellum continued to expand to manage balance, coordination, and timing. Each movement demanded awareness of limb placement, branch stability, and the location of nearby group members. This constant need for coordination strengthened neural pathways between sensory regions and motor centers, enhancing both precision and adaptability. The dietary diversity of early primates also played a role in their neural evolution. Many species shifted to omnivorous diets, eating fruit, leaves, insects, and occasionally small animals. Finding and processing these foods encouraged exploratory behavior and problem solving. Fruits ripen at different times, insects hide in small spaces, and edible plants grow unevenly across forests. These challenges required memory of food locations, timing, and flavors, further enriching cognitive development. Over time, early primates experienced selective pressures that favored individuals with stronger memory, better coordination, and more effective social strategies. These traits helped them survive periods of scarcity, avoid predators, and maintain group cohesion. As generations passed, the neocortex expanded further, forming dense networks of neurons that supported increasingly complex thought. In the gentle sweep of this evolutionary chapter, we see how early primates laid the groundwork for advanced cognition and social connection. Their brains became webs of interlinked systems, sensory, emotional, motor, and cognitive, all communicating in harmony. These animals learned to navigate not only the physical environment but also the intricate social landscapes of group living. From these early primates arose the ancestors of monkeys, apes, and eventually humans. The architecture of social minds, created through millions of years of adaptation, would carry forward into new environments, new challenges, and new possibilities. The story of the brain was entering a stage defined not only by survival, but by communication, cooperation, and the beginnings of a deeper awareness elements that would shape the next great chapters of evolution. 17 early humans and the slow growth of intelligence. In the long and unbroken flow of evolutionary time, after the primates had already shaped minds capable of social awareness, careful movement, and rich sensory perception, the Earth began offering new landscapes and new challenges. Climate cycles shifted, forests receded in some regions, and open grasslands spread across the horizons of ancient continents. Within these changing environments, a particular lineage of primates began to walk more frequently on two legs, gazing across vast distances and exploring unfamiliar territories. From these subtle changes emerged the earliest humans creatures whose brains were beginning to take on new forms of organization, preparing them for futures shaped by curiosity, cooperation, and a quiet but growing intelligence. To imagine these early humans is to picture small groups moving together through patches of forest and open plains. Their bodies were lean and strong, adapted for long-distance travel, their hands free to carry tools or food. Unlike many earlier primates, they walked upright for much of their lives, a shift that changed the demands placed on their nervous systems. Walking on two legs required constant, fine adjustments in balance and coordination. The cerebellum worked in gentle partnership with the spinal cord and motor cortex to maintain stability with each step. A subtle variation in terrain, a stone, a slope, a patch of tall grass required immediate neural interpretation and adjustment. This new posture also changed the relationship between the body and the world. With their hands freed from locomotion, early humans could manipulate objects more precisely than their ancestors. Simple tools, at first little more than stones broken into sharp fragments, began to appear. These tools required planning. The brain had to imagine their shape, anticipate their use, and remember the steps involved in creating them. Even the earliest stone flakes represented a small but profound leap in cognition. The motor cortex refined its control over hand movements. The sensory cortex processed information from touch in new ways, and the frontal lobes began to support more advanced problem solving. Tool use reinforced the growth of neural pathways that guided behavior beyond instinct. When early humans shaped a stone tool, they engaged in a kind of quiet experimentation. They learned which stones were stronger, which angles produced sharper edges, and which motions created the desired result. These lessons were stored not only in muscle memory. The brain grew better at linking sensory experiences with outcomes, strengthening circuits that supported learning and planning. Over generations, this skill became a defining trait of human ancestors. At the same time, changes in the environment encouraged new patterns of cooperation. Early human groups foraged together, protected one another, and raised young in shared communities. These social bonds required the brain to track relationships, understand intentions, and respond to emotional cues. The limbic system, especially regions associated with attachment and social behavior, grew more influential. Individuals became more attuned to one another's needs. These early forms of social communication did not yet involve language, but they laid the groundwork for the more complex interactions that would come later. Memory also deepened during this period. Early humans needed to remember the locations of food sources, seasonal migration routes, and shelters that offered safety. The hippocampus, already significant in earlier primates, developed richer connections with the expanding neocortex. Behavioral memory allowed individuals to recall strategies that had succeeded in the past. Together, these forms of memory supported survival in landscapes where resources shifted with weather and migration patterns. As diets expanded to include more diverse foods, including meat found through scavenging or simple hunting, the nutritional demands of the brain were met with greater efficiency. Foods rich in fat and protein supported neural growth, allowing the brain to expand gradually in size and complexity. This nourishment likely influenced both the structure and the function of the nervous system, supporting the development of more intricate neural networks. With each generation, the brain gained new layers of connectivity, allowing early humans to think more flexibly and solve problems with greater nuance. Communication among early humans grew more sophisticated as well. While full language did not yet exist, vocalizations became more varied. Different sounds conveyed different emotional states or intentions. Simple calls warned of danger, signaled food. These vocalizations required coordination between auditory regions, motor pathways, and the emerging structures in the frontal cortex that would one day support language. Early humans also communicated through gestures, signals of reassurance, direction, or invitation, which further strengthened the brain's ability to interpret others' actions and intentions. Another quiet transformation occurred in the way early humans experienced time and anticipation. Their growing cognitive abilities allowed them to think ahead to foresee the benefits of storing food, to anticipate seasonal changes, or to prepare tools before they were needed. These abilities reflect early forms of executive functioning, supported by expansions in the prefrontal cortex. This region helped early humans plan, evaluate outcomes, and delay immediate impulses in favor of future benefits. These skills would become essential as human societies grew more complex. The emotional lives of early humans also deepened, caring for infants, cooperating during hunts, grieving losses, and celebrating small successes strengthened social bonds. Hormonal systems worked in harmony with neural circuits to reinforce these behaviors. As emphasized in your uploaded materials, the interaction between nervous, endocrine, and sensory systems forms the basis for complex biological functions. In early humans, This integration supported emerging forms of empathy, attachment, and shared intention traits that would later become hallmarks of human behavior. As early humans crafted tools, explored landscapes, and interacted with one another in increasingly complex ways, the shape of their brains began to reflect their expanding capabilities. The neocortex grew larger relative to body size, forming new folds and layers that supported flexible learning and decision-making. The parietal regions strengthened their role in interpreting spatial relationships, while the temporal regions gained new capacities for processing sound and memory. The frontal lobes expanded their influence, guiding behavior with foresight rather than pure reflex. Yet despite these advancements, the growth of intelligence was gradual and gentle, shaped by countless generations of small adjustments. Early humans did not suddenly become capable of abstract thought or symbolic communication. Instead, their minds expanded slowly, shaped by the rhythms of their environment, the demands of cooperation, and the quiet challenges of survival. Their evolution was not a story of sudden leaps, but a long sequence of small, harmonious changes that enriched the brain's architecture. In this soft and steady progression, the seeds of future human cognition were planted. The ability to plan, remember, cooperate, and interpret emotion all emerged in these early ancestors. Their brains carried forward the wisdom of millions of years of vertebrate evolution, while adding new layers of adaptability that would eventually support language, culture, and creativity. As we reflect gently on this stage of the journey, we can see how early humans began bridging the gap between instinct and understanding. Their minds were becoming landscapes of memory and possibility, capable of shaping tools, forming bonds, and exploring new worlds. The slow growth of intelligence, quiet yet profound, set the trajectory for the extraordinary cognitive development that would follow, guiding evolution toward the intricate and reflective minds of the modern human lineage. 18. The modern human brain, a soft anatomy. In the long and gentle arc of evolution, after early humans had begun shaping tools, forming memories, and nurturing deeper social bonds, the human brain continued to grow into a structure of extraordinary subtlety. This growth was not sudden, nor was it dramatic. It was a quiet accumulation of refinements each generation, adding a little more complexity, a little more connectivity, and a little more nuance to the architecture that had been forming for hundreds of millions of years. By the time anatomically modern humans appeared, the brain had become a landscape of remarkable intricacy, filled with layers of memory, emotion, movement, and thought woven together into a single, softly coordinated whole. To explore the modern human brain is to walk through an ancient forest of structures, each one shaped by different chapters of evolutionary history. Some regions are old and instinctive, guiding basic survival behaviors inherited from reptiles and early mammals. Others are newer, supporting imagination, language, and reasoning. All of these regions work together harmoniously, exchanging signals through elaborate networks of neurons that fire in patterns as delicate as the flicker of candlelight. The outermost layer of the brain, known as the cerebral cortex, is perhaps its most recognizable feature. This gently folded sheet of neural tissue, only a few millimeters thick, is responsible for many of the functions that define the human experience. Its surface is shaped by ridges and grooves that increase the available area for neurons, allowing more connections and more complex processing. Within this layer lie regions devoted to movement, sensation, hearing, vision, language, planning, and abstract thought. Each region communicates continuously with others, forming a dynamic map of perception and behavior. The frontal lobes, nestled behind the forehead, are among the most expanded regions in humans. Within the frontal lobes lies the prefrontal cortex, a structure that helps humans imagine the future, weigh options, and choose among possible paths. It is also involved in emotional regulation, linking deeply with the limbic system to help shape responses that are thoughtful rather than instinctive. This ability to pause, reflect, and choose is one of the hallmarks of the human mind. Nearby, the motor cortex plays its role in guiding movement, Each small region of this cortex corresponds to specific muscles of the body, coordinating fine and precise motions. These motor signals travel down through the spinal cord in orderly pathways, activating muscles with graceful timing. Adjacent to the motor cortex lies the somatosensory cortex, which receives touch information from the skin. This region forms a quiet internal map of the body, allowing humans to feel pressure, warmth, and texture with clarity. This organization, as described in your uploaded materials, reflects the nervous system's role in gathering information, integrating it, and coordinating responses throughout the body. Further back, the parietal lobes help the mind interpret space and motion. These regions weave together information from the eyes, ears, and skin. They help humans reach for objects, walk smoothly, and navigate surroundings. The parietal lobes also play a role in numerical reasoning and in the integration of sensory maps, quietly supporting both everyday tasks and more abstract cognitive abilities. The occipital lobes, located at the back of the brain, are dedicated to vision. Here, patterns of light become meaningful shapes, colors, and movements. Lines are recognized, faces are perceived, and complex scenes are interpreted. This region communicates constantly with the temporal and parietal lobes, blending perception with memory and context. On the sides of the brain, the temporal lobes contribute to memory, language, comprehension, and sound processing. These regions help humans understand speech, interpret complex auditory patterns, and recall memories. Deep within the temporal lobes lie the hippocampi, small, seahorse-shaped structures essential for forming new memories. They act as the brain's navigators, mapping experiences in both time and space. These structures, which began their evolutionary journey, helping reptiles and mammals navigate simple terrain, now support the richly layered memories that shape human identity. Interwoven with these cortical regions lies the limbic system, a network of structures associated with emotion, memory, and instinct. The amygdala, one of its key components, helps interpret emotional significance, fear, joy, surprise, and subtle social cues. It works closely with the hippocampus, linking memories to emotional states. These interactions subtly guide human behavior, encouraging caution when recalling danger, or comfort when remembering safety. Though the limbic system predates many cortical structures, it remains deeply integrated with the neocortex, shaping thoughts with emotional tones. Beneath the cortex, the brainstem supports life's essential rhythms. It regulates breathing, heart rate, sleep cycles, and reflexes. It acts as the quiet foundation upon which all other brain activity rests, maintaining the internal balance that supports consciousness. The brainstem interacts with the cerebellum, a structure tucked beneath the occipital lobes, famous for its role in balance, timing. The cerebellum fine-tunes movements so they become smooth and graceful. The cerebellum's quiet calculations highlight an important truth. The brain is not a collection of isolated parts, but a symphony of interconnected systems. Signals travel constantly between regions, shaping perception, emotion, memory, and action into a seamless experience. Neurons communicate with electrical and chemical signals, forming vast networks that learn, adapt, and reorganize as life unfolds. These signals reflect the nervous system's foundational role described in your materials, processing information, coordinating responses, and maintaining the body's internal equilibrium. Glial cells, often overshadowed by neurons, support this complex ecosystem. They nourish neurons, maintain chemical balance, insulate axons, and help guide neural development. Together, neurons and glia form the living threads of cognition and perception. Structures involved in interpreting facial expressions, understanding others' intentions, and empathizing with emotional states are highly developed. This social sensitivity is part of what makes human societies possible. By reading subtle cues in voice and posture, humans navigate relationships, build trust, and maintain communities. Language is another defining feature. The brain regions responsible for language production and comprehension, primarily in the left hemisphere, for most individuals enable humans to share thoughts, express memories, and build culture. These regions connect deeply with auditory areas, motor pathways, and associative networks, allowing fluid expression through speech, writing, and gesture. As the human brain evolved, it did not discard older structures. Instead, it built upon them, layering new functions atop ancient foundations. Instinct and emotion remain deeply embedded, yet they are shaped and refined by higher-order cognition. Memory is woven from both factual recall and emotional resonance. Movement arises from a partnership between conscious intention and automatic coordination. When viewed gently, the human brain appears not as a static machine, but as a living map of evolutionary history, a sanctuary of sensations, feelings, and ideas. It holds the wisdom of ancient oceans, the alertness of reptiles, the emotional warmth of mammals, the social intelligence of primates, and the reflective depth of early humans. It is a landscape shaped by time, carrying within it all the quiet transformations that allow human beings to perceive the world, navigate relationships, and create meaning. As this part of the journey closes, we remain aware that the modern brain, though exquisitely complex, is not an endpoint. It is the current expression of an ongoing story, a story that continues to unfold with each generation, each experience, and each quiet moment of thought. 19 neural networks, memory, and the flow of thought. In the serene continuity of the human story, once the brain had settled into its modern anatomical form, with its folded cortex, its deep limbic layers, and its rhythmic brainstem, another transformation began to unfold, one that did not alter the brain's physical shape, but rather the way it functioned moment to moment. This transformation was rooted in the intricate web of networks inside the brain, networks that made it possible for thoughts to flow, memories to take shape, and experiences to weave themselves into the quiet tapestry of awareness. In understanding these networks, we can sense the living architecture of the human mind, an architecture defined not by static structures alone, but by the continual movement of signals traveling softly through billions of interconnected neurons. Each neuron in the brain is a small but extraordinary cell. It receives information, processes it, and passes it onward, contributing to the delicate patterns that form perception, memory, and thought. The nervous system, described in your uploaded materials as the body's communication framework, works by transmitting electrical impulses along neurons and chemical signals across the spaces between them. These transmissions form the true foundation of experience, a moment of recognition, a feeling of comfort, a memory drifting to the surface. Each one arises from patterns of signaling that ripple through neural networks with gentle precision. At the core of these networks lies the synapse, a small gap between neurons, where chemical messengers, known as neurotransmitters, carry signals from one cell to another. The strength of these synapses changes over time, subtly shifting based on experience and learning. Some connections grow stronger with repeated use, while others grow softer and fade. This dynamic quality gives the brain its remarkable plasticity, the ability to adapt, reshape itself, and form new pathways in response to life's changing demands. Memory, in its many forms, emerges from this plasticity. It is not stored in a single place, nor does it exist as a fixed imprint. Instead, it is woven through networks of neurons that fire together in repeating patterns. When a person relives a memory, the brain reactivates the network that created it, drawing from sensory regions, emotional centers, and associative pathways. Some memories are vivid and rich with detail, while others are softer impressions that fade gently into the background. This variation reflects the different kinds of memory the brain holds. Short-term memory, for example, is like a small pool that holds information briefly before releasing it. It allows a person to keep a thought in mind for a moment, perhaps a spoken sentence, a number, or a familiar face, before passing it on to deeper circuits for storage. Working memory, another form, supports problem solving and reasoning, holding and manipulating information in the moment. Long-term memory arises when certain neural patterns are reinforced strongly enough to become enduring. These memories can be episodic, tied to personal experiences, or semantic, holding general knowledge about the world. They can also be procedural, stored in motor pathways that help the body perform practice gestures like walking. The hippocampus plays a central role in forming new long-term memories, guiding them towards storage in the neocortex, where they settle into the broader network of knowledge and experience. The limbic system, with its emotional centers, shapes memory in a special way. Memories associated with strong emotions tend to be retained more vividly. The amygdala, for instance, helps attach emotional significance to events, making certain moments easier to recall. This integration of emotion and memory has deep evolutionary roots. In early vertebrates, emotional reactions served as guides for survival, prompting caution, attraction, or avoidance. In humans, these emotional traces enrich memory, giving it meaning and shaping how experiences are interpreted. Thought itself arises from the interplay of multiple networks. It is not located in one region of the brain, but instead emerges from the interactions between sensory processing areas, association cortices, memory pathways, and the frontal lobes. The prefrontal cortex, in particular, acts as a conductor, coordinating attention, decision-making, and planning. It helps organize thoughts into coherent sequences, evaluates possibilities, and selects actions. This region also helps regulate emotional responses by communicating with deeper limbic structures, guiding choices in ways that reflect both instinct and reflection. Speech and language are similarly networked processes. They rely on regions responsible for understanding voice, generating words, coordinating breath, and shaping sound. These regions work in harmony with memory systems, allowing people to recall vocabulary, understand grammar, and connect language with meaning. The networks supporting language are highly interconnected, drawing on the auditory cortex, motor pathways, and multiple association regions of the cortex. Another important aspect of neural networks is the default mode network, a system active when the mind is at rest, daydreaming, or reflecting inwardly. This network supports introspection, imagination, and the soft unfolding of thoughts. It activates when external demands quiet down, allowing the mind to wander through memories, possibilities, and inner landscapes. This inward flow of thought connects past experiences with present perspectives, allowing meaning and understanding to deepen over time. Attention networks help the mind focus on the present moment, guiding perception toward relevant information. Emotional networks color thoughts with subtle tones of feeling. Memory networks bring context. Together, these systems guide awareness gently from one moment to the next. If one network becomes overly active or underactive, thought patterns may shift. Yet even then, the brain's plasticity allows it to adjust over time, seeking a new equilibrium. Throughout the day, the brain remains in continual dialogue with the body. Signals from the nervous system integrate with those from the endocrine system, regulating mood, energy, and alertness. As noted in your materials, these systems work together to maintain balance and support the body's needs. Hormones influence neural activity, shaping motivation, memory formation, and emotional states. This interplay adds depth to human experience, blending physical sensations with cognitive and emotional responses. As the day ends and sleep begins to settle in, the neural networks shift once again. During sleep, especially in the deeper stages, the brain consolidates memories, strengthens important connections, and softens those that are no longer needed. This nightly restoration keeps the mind flexible and clear, allowing thought and learning to flow naturally the next day. In the soft understanding of these processes, one can see that the brain is not merely an organ of thought, but a living landscape shaped by experience, emotion, and connection. Its networks are fluid, always adapting, always responding to the subtle movements of life. The flow of thought is not linear or mechanical, it is gentle, rhythmic, and deeply intertwined with memory, sensation, and feeling. As modern humans move through their days, they carry within them this dynamic web of connections, an intricate system shaped by millions of years of evolution, yet uniquely molded by the quiet experiences of each individual life. These networks allow people to learn, remember, imagine, and dream. They allow the mind to explore inner and outer worlds with grace and curiosity, and they stand as one of the most delicate and remarkable expressions of biological organization ever to arise on Earth. In this soft expanse of thought and memory, the modern human brain continues to reveal the depth of its quiet intelligence. Its networks form the foundation for consciousness, creativity, and reflection traits that shape not only individual lives, but the shared journey of humanity as a whole. 20. The continuing evolution of the human mind. In the gentle sweep of time, after the human brain had taken its familiar form with its layered memories, flowing thoughts, and intricate sensory worlds, its evolution did not come to a sudden halt. Instead, it entered a quieter, more subtle phase of change, shaped not only by biology, but by culture, experience, and the shared patterns of human life. While the physical structures of the brain remained largely stable across many thousands of years, the way these structures were used continued to transform. Human minds adapted to new environments, new tools, new relationships, and new ways of understanding the world. The story of the brain became not only a biological narrative, but also a cultural one, shaped by learning, language, art, and the steady passing of knowledge through generations. In this modern chapter of evolution, one of the most significant forces shaping the brain is its own capacity for learning. Humans are born with brains that are highly plastic, able to form new connections, strengthen useful pathways, and gradually refine themselves through experience. Each day, the brain receives countless signals from the body and the environment. Some of these signals are woven gently into memory, while others fade. This continuous conversation between experience and neural structure allows the mind to grow throughout life. Even in adulthood, the brain remains capable of forming new memories and reorganizing itself around new skills or challenges. This adaptability reflects the three-step neural process described in your uploaded materials: sensory input, integration, and motor output, a cycle that shapes both behavior and understanding. Language has played a central role in this ongoing evolution. The ability to share ideas, experiences, and emotions through speech and writing created a new kind of inheritance, one that traveled not through genes, but through words, stories, and shared symbols. Language allowed human groups to teach one another, coordinate actions, settle conflicts, and build traditions. Over time, these cultural exchanges encouraged the brain to develop stronger associative networks. The temporal lobes, involved in language comprehension, communicated with frontal regions responsible for planning and reasoning. This collaboration enriched thought, allowing people to form abstract concepts, imagine possibilities, and create systems of knowledge that extended far beyond immediate experience. Culture also shaped memory. As societies grew more complex, humans began to store collective knowledge, first through oral traditions, then through writing, and eventually through vast networks of shared information. These cultural practices reflected the brain's natural tendencies to observe, interpret, and pass on information. When a story was told around a fire, or when a piece of wisdom was taught from one generation to another, the brain reorganized itself to reflect these patterns. Over time, this interplay between culture and biology created layers of understanding that became part of the human experience. The brain's emotional landscape also continued to evolve. Humans formed deeper bonds with one another, guided by neural circuits that had been shaped long before in early mammals. Social structures grew more complex, requiring individuals to interpret subtle cues, manage relationships, and cooperate in increasingly intricate ways. The limbic system worked closely with the prefrontal cortex to regulate emotions, encouraging thoughtful responses rather than instinctive reactions. This collaboration helped humans live in large communities, care for one another, and share responsibilities. Creativity blossomed as well. With the expansion of the neocortex, humans developed the ability to imagine worlds that did not yet exist, to create music, art, stories, and tools. These acts of creativity were expressions of neural networks working in harmony, visual regions, collaborating with emotional centers, memory systems linking with motor pathways. This interplay allowed people to shape their environments, build shelters, craft instruments, and adorn their surroundings with patterns and symbols that carried meaning. The brain also adapted to new forms of problem solving. Early humans had needed to navigate natural landscapes, find food, and avoid danger. Modern humans face different challenges designing tools, understanding weather patterns, cultivating plants, and building communities. These new demands encourage the brain to refine its ability to analyze patterns, predict outcomes, and draw connections between seemingly unrelated ideas. The prefrontal cortex became increasingly important in supporting these abilities, guiding both individual decisions and collective actions. Social cooperation grew into one of the defining features of human life. The brain's areas for empathy, perspective taking, and emotional regulation allowed people to understand one another's needs and feelings. This understanding supported teamwork, conflict resolution, and shared goals. As communities expanded, these social abilities became essential for survival. Human groups relied on one another for food, protection, parenting, and cultural transmission. The brain evolved not only to engage with the physical world, but also to navigate the complex terrain of relationships and collective identity. With the passage of time, humans developed the ability to reflect on their own thoughts, a form of introspection supported by the brain's default mode network. This network, which becomes active during quiet moments, allows people to consider their memories, imagine the future, and contemplate their place in the world. It provides space for reflection, creativity, and self-understanding. This introspective capacity deepened the human experience, allowing individuals to form personal narratives and explore meaning in their lives. The brain's ongoing evolution also intertwines with technology. Even though the biological structure remains largely the same, the ways humans interact with the world continue to evolve. Tools, instruments, and systems of communication shape patterns of thought, encouraging new forms of memory, attention, and creativity. The brain adapts to these tools through its natural plasticity, forming new pathways and connections to support new skills. Throughout all these changes, the brain retains its ancient foundations. Beneath the complexity of modern thought lie circuits shaped by early vertebrates, reptiles, mammals, and primates. The brainstem continues to guide breathing, heart rate, and basic rhythms. The limbic system continues to color thought with emotion. The neocortex continues to weave perception, memory, and imagination into coherent experience. These layers work together, blending instinct, emotion, and reason into a unified whole. In this way, the human mind remains a living reflection of its evolutionary history. It carries the adaptability of early fish, the sensory refinement of reptiles and birds, the emotional depth of mammals, and the social intelligence of primates. It builds upon these foundations with the unique capacities of modern humans: language, creativity, introspection, and culture. The mind is not a static structure, but a gentle flow of networks, memories, and meanings shaped by both biology and experience. As we arrive at the final chapter of this journey, the story of the brain continues to unfold. Each generation inherits not only the biological framework of the mind, but also the cultural and experiential patterns passed on through learning. The brain remains open, flexible, and capable of growth throughout life, adapting to new challenges with quiet resilience. In this soft understanding, we see that the evolution of the brain is not simply a story of structures and neurons, it is a story of experience, cooperation, creativity, and reflection. It is the story of how life learned to sense the world, to remember it, to imagine it, and to shape it. And as long as humans continue to think, feel, and dream, the journey of the mind will remain an ever unfolding chapter in the long and gentle history of life on Earth.